माइक ऑन है नहीं, माइक ऑन, साउंड ऑफ। मैडम आरेक बार कुक करते हैं।
Members of the faculty, staff, scholars, and the young friends and their mentors who have joined us today from various colleges across West Bengal. It is a great honor and pleasure to welcome you all on this day, the 28th of February, when we, along with the entire nation, are all set to celebrate National Science Day 2024, which not only commemorates the discovery of the Raman effect by Sir Chandrasekhara Venkata Raman, but it is also a day which provides an opportunity to spread the message of the importance of science and its application in our lives, to encourage the youth to engage in scientific activities and careers in science and technology, and inculcate scientific temper and foster a culture of innovation among the general public. This year's celebration of National Science Day is based on the theme of Indigenous Technologies for Viksit Bharat, which emphasizes the importance of science in making India Atmanirbhar and reaffirms the vision of Amrit Kal, Viksit Bharat at 2047. The theme reflects the increased focus of the government on the indigenization of technologies. May I now kindly request our honorable guest, Professor K. Thangaraj, the director, to kindly come up on the dais, please. And I would request the chairperson of the organizing committee, Professor Deboshree Ghosh, to kindly accompany them. I would now request our PhD students, Ms. Payal Shatra and Ms. Onushmita Ghosh, to kindly felicitate our honorable guest, Professor Thangaraj, firstly with a bouquet and then with a replica Raman spectrometer as a token of remembrance from IACS. Next, we shall be felicitating our director, Dr. Rangan. And last but not least, Professor Deboshree Ghosh. May I now kindly request the chairperson of the organizing committee, Professor Deboshree Ghosh, to kindly present the welcome address.
Good morning, all of you. On behalf of the Indian Association for the Cultivation of Science, I welcome you all to the celebration of the National Science Day. As many of you know, the National Science Day is celebrated every year on the 28th of February to commemorate the discovery of Raman effect by Sir C.V. Raman. He announced his path-breaking discovery on this day in 1928. For this discovery, Sir C.V. Raman was awarded the Nobel Prize in Physics in 1930. And the government of India has designated this 28th of February as the National Science Day starting from 1986. Since then, National Science Day is being celebrated every year throughout the country through public lectures and various other theme-based activities. The theme for the National Science Day 2024, as was mentioned before, is Indigenous Technologies for Viksit Bharat. The theme highlights that the way to an abundant and prosperous country is rooted in the development of science and technology. The accomplishments of many scientific ideas and breakthroughs and the benefits of science and technology should reach every household and bring ease of living to common people in our country. Here in IACS, we have put together an exciting program particularly for the students that includes lectures by eminent scientists on a broad range of topics that highlights these laudable indigenous endeavors and technologies. We also have a lab visit that is scheduled after this in the afternoon session. The topic of the discussion ranges from biology to physics. We have a lecture that will discuss about our health and diseases. We have a lecture that includes emergent devices and next generation technologies. The lectures will encompass, as you can see, a variety of topics, but will definitely give a glimpse of the curiosity and experimentation that leads to enterprise and products. I hope you will appreciate that science is not just about a set of subjects like maths, physics, chemistry, biology, etc. It is more important to embrace the scientific temperament, scientific and rational way of thinking in our daily lives. In my view, that is how we should try to celebrate Science Day, and every day should then become a Science Day. I hope you will enjoy the rest of the day. I would invite all of you to actively participate and interact with the speakers during your stay. Thank you.
May I now kindly request our director, Dr. Ranjan Sen, to kindly present the inaugural address, please, sir.
Good morning, everyone. My dear faculty, staff, and my beloved students, and also their teachers who came with them. As we know that today we gather to commemorate a remarkable occasion that transcends border, ideologies, and cultures. It's called National Science Day. It is a day of reflection, appreciation, and anticipation for science's profound impact on our lives. On this day, we celebrate the remarkable discovery made by Sir C.V. Raman, one of the greatest scientists of our country, who brought India to the global forefront of scientific excellence. His discovery, famously known as the Raman effect, really changed the field of physics, opening new avenues for scientific inquiry and technological innovations. Sir C.V. Raman and Dr. K.S. Krishnan's seminal paper on Raman effect was published as a letter to the editor in Nature on February 18, 1928. It was a brief communication that introduced the groundbreaking discovery of the scattering of light by light molecules, which became known as Raman effect. The discovery was announced on 28th February 1928. So that's the start. C.V. Raman was closely associated with IACS most of the major part of his career. His association with the institute began early in his professional life. Many people don't know this history, that in 1907, C.V. Raman actually joined Indian Finance Department as an assistant account general, which brought him to Kolkata. And his office was very close to the then IACS campus building at Beaubazar. So he used to come very frequently, and during this time, he became engaged with the scientific community of IACS as well as the scholars of CT. In 1917, C.V. Raman was appointed as the Palit Professor of Physics at the University of Calcutta by then Vice Chancellor Ashutosh Mukherjee, Calcutta University Vice Chancellor, a position that was closely linked with IACS, where he can perform his research work. While at the university, Raman continued his research and collaboration with the scientists at the IACS, laying the groundwork for the future discoveries. One of the most notable periods of Raman's association with the IACS came during 1920s, when he conducted the experiments that led to the discovery of Raman effect in 1928, and he received the Nobel Prize in Physics in 1930 for this discovery. Now, what is Raman effect? The Raman effect is a phenomenon in spectroscopy when light scattered by a molecule undergoes a frequency shift. The frequency shift, known as Raman scattering, occurs when incident light interacts with the vibrational modes of the molecules. When light interacts with a molecule, most of the scattered light maintains the same frequency as the incident light, which is very well known as Rayleigh scattering. However, a small fraction of the scattered light experiences changes in the frequency due to the interactions with the vibrational and rotational states of the molecule. These frequency shifts correspond to the energy differences between the initial and final states of the molecular vibration and rotational modes, and that we see as a Raman scattering. Raman scattering has a lot of applications. It provides valuable information about the molecular structure, chemical bonding, vibrational modes of the substances. It allows scientists to analyze identify chemical compounds, study molecular dynamics, and investigate the properties of the material. These are measured and studied by Raman spectroscopy based on the Raman effect. It has become a powerful analytical tool used in the various fields, including chemistry, physics, biology, material science, and pharmaceuticals. Now, the history of National Science Day traces back to 1986, when the Indian government decided to designate February 28th as the National Science Day. The inaugural National Science Day was celebrated in 1987 with various events, seminars, lectures, and science exhibitions organized nationwide to promote scientific awareness and foster a scientific temperament among the masses. Since then, National Science Day has become an annual celebration that brings together scientists, researchers, educators, and most importantly, the students, the budding scientists, and the general public to reflect on the importance of science in the society and to showcase India's achievement in the field of science and technology. The theme for National Science Day, this year's National Science Day in 2024 is Indigenous Technologies for Viksit Bharat. Only technological innovation will lead us to become a $1 trillion economy and place us as the world's leader. National Science Day serves as a reminder for the transformative power of science in shaping our world. From unraveling the mysteries of the cosmos to the decoding the complexities of human genome, science has been our guiding light, illuminating the path towards progress and prosperity. Scientific discoveries have enriched our lives in myriad ways, from the advancements in medicine, technology, to conserving our environment, natural resources. This day also celebrates the enduring spirit of scientific inquiry. It is this spirit that drives researchers and innovators to push the boundaries of the knowledge, question the unknown, seek solutions to the most pressing problems of our time. It is this spirit that unites us in our shared pursuit of a better tomorrow, a tomorrow defined by progress, prosperity, and sustainable development for all. So, let us march forward with the determination and optimism, guided by the light of reason and promise of a brighter future for all. Thank you very much. Now I will introduce our today's first speaker, very distinguished scientist, Dr. Kumaraswamy Thangaraj. Actually, I am very fortunate to work under his leadership. He was a director for 3 years in my previous institute, Center for DNA Fingerprinting and Diagnostics in Hyderabad. And he is a very well-known human geneticist. I will just briefly, and you will be hearing a very exciting talk today, especially for the students, like how our genetic makeup controls our disease state and why we get certain disease, why we don't get certain disease, why we are different from other animals, why our immune systems are different. So it's all written in the genetic code, and that's a study, and he will be talking about that. He will give a very interesting examples today, and I'm sure all of you will be enjoying. But let me give a very brief CV about him. He's quite well-known person. Dr. Kumaraswamy Thangaraj is now a J.C. Bose Fellow at the CSIR Center for Cellular and Molecular Biology, CCMB, which, those who have gone to Hyderabad, especially for the students. So there is a eastern part of Hyderabad, there is a cluster of research institutes. One of them is CCMB, next to it then CDFD, that where I used to be there. And there is another institute called NIN, IACS, so all are in one place. It's a very beautiful academic hub. And he was the director of the Center for DNA Fingerprinting and Diagnostics, Hyderabad, from 2020 to 2023. He was also director, additional chair to Institute of Stem Cell Science and Regenerative, that is called NIAB, in STEM, so that is in Bangalore. He was And his main research for the last decades, mostly in the field of population and medical genomics. He was published about 300 research articles. He is an elected fellow of all the major science academies in India. He is a recipient of several awards, like including the J.C. Bose Fellow, Sun Pharma Research Award, award in Raman Research Fellowship, Distinguished Scientist Awards, a C.V. Raman Memorial Lecture Award, and Sir Dr. U.N. Brahmachari Award, and many. And he also delivered many several orations. He is a board of editor of Mitochondria, associate editor of BMC Medical Genetics, Tropical Medicine International Health, and editorial member of Human Genetics and Clinical Genetics. He was the president of Indian Society of Human Genetics from 2011 to 2015 and founder of the Society for the Medical Mitochondrial Research and Medicine. So the title of the talk is Who Are We and What Makes Our Health and Disease So Unique? Please enjoy his talk.
Very good morning to all of you, kids. I thank Dr. Ranjan Sen, director of this institute, and also my friend and colleagues for inviting me to talk on this occasion. In fact, it was my pleasure and privilege to speak on this occasion where Dr. Sir C.V. Raman was associated with this particular institute. I also thank the chairperson and colleagues of this great institution. So what I am going to talk to you is about our own origin, right? I will not go into very detail, but take you through some journey, my own research, of course towards end, and how that had helped us in understanding who are we. It is not a philosophical question, but this is a question everybody thinks, and people are more interested in knowing about their own origin in many continents. But also important that knowing our own origin also helps us in understanding our health and disease. Right? Before going into this, one would realize after seeing this, so why we have to understand evolution. Dybzhansky, he was a professor in Rockefeller University. He wrote an article which was published in the journal called the American Biology Teacher in 1973, where he has mentioned that nothing in biology makes sense except in the light of evolution. You will really understand that as my talk go along, that you will realize that how important this statement is. You are all well aware of Charles Darwin, who is a father of evolution, and he tried to understand the human evolution, how the human or Homo sapien has evolved. But he wrote a book in 1858, it's called as The Origin of Species, right, where he has mentioned about origin of many organisms, right from arch to whales. But he could not imagine that how human might have originated. So that was missing in this particular book. There is another evolutionary biologist, Thomas Henry Huxley. He wrote a book called Evidence as to Man's Place in Nature. Using anatomical similarity, he predicted that there was evidence that human and ape must have come from a common ancestor. Several years later, Darwin wrote another book called The Descent of Man, where he has declared that chimpanzee and gorilla are our closest living relatives based on the anatomical similarities. So, that was the first time these two great evolutionary biologists predicted that probably we must have originated from ape or chimpanzee. But today, with the human genome sequence and sequencing of many other organisms, it is more clear that we, the human, genetically more closer to chimpanzee, and it gave evidence that human and chimpanzee have diverged from a common ancestor called hominids about 6 to 7 million years. So, how many of you believe that we have come, we in the sense human and chimpanzee have come from a common ancestor? Anybody say no? Right? But in the process of evolution, genetics played a major role. Whatever the DNA change, that will bring a new phenotypic perspective or some disease or adaptation, right? All of this happens because of the change in our DNA. So, if you imagine that we all have come from a common ancestor, if you do not believe on this, you look at these three brothers who born to a normal parents, right? There was a change in the DNA which brought feature like the way we look like today. But they born to a normal parents, the parents were absolutely fine. But then why they look like chimpanzee? It's because probably there is a back mutation. The mutation we gained which gave us the feature like the way we look like today. It has gone back, therefore, they appear like chimpanzee like features. There are some more evidence. Look at this girl. This is one of the features we lost, the body full of air, the course of evolution. Again, there is a genetics behind that. But there is a back mutation which probably brought body full of air. It is unfortunate for this young girl. We have one more evidence. This came very recently. You can go into YouTube and look at this video very recently. This is a Turkish family. There are 5 daughters and 1 son. So, only the elder son walked with 2 legs, but not again steadily as a drunken fellow. He cannot walk steadily. But 5 sisters, they walk with 4. That means this is again one of the features we gained during the course of evolution. Again, there is a mutation in the gene which helped us in walking with the 2 legs. There is a back mutation. Therefore, the 5 sisters, you can see the video actually, they walk with 4. So, all of this gives evidence that definitely we all come from a common ancestor. There is a mutation which helped us in evolving like the way we look like today. So, it is not happened suddenly. There is a mutation from chimpanzee, we turn into human. There are several species. I told about 7 million years. This is 0, 1, 2, 3 million, 4, 5, 6 and 7 million. I mentioned that one of the ability is walking with 2 legs, right. Another ability is making tools for the survival, killing animal or hunting for food. The third important thing is our brain size has increased. If you look at the early species chimpanzees, the brain size was as similar as chimpanzee. Okay. Then it has increased to 35% of the modern human. Then in case of Homo habilis, 50% increased to 60%. Interestingly, in Homo neanderthals, they are our most closest in terms of evolution. They had slightly larger brain than human, but one can imagine whether they are more intelligent than human Nobody knows. But why that species have vanished? Again, nobody knows. The human evolved and they actually removed all other species but the closest species. That is what one can predict. Interestingly, one of the evolutionary biologists from Cambridge, he said the inherent violent behavior of modern human, we all see, right, how violent we are, responsible for vanishing of Neanderthals. This could be true. But when we talk about Neanderthals, it is very, very important in the context of modern human evolution that was found in Europe, different part of the Europe. You can see there is a Neanderthal valley. That is where it was found for the first time. And there was a lot of ancient DNA work has been done by Svante Pääbo. This was the first time Svante Pääbo has sequenced 500,000-year-old the Neanderthal bones, and he gave actually the nice evolutionary connection between human and the hominids. Okay. And because of that, has been awarded with Nobel Prize last year, the field of medicine and physiology. When we talk about Neanderthal and human, because it gave a very good connection, we looked at 300 human genome from different part of the world, which I will show you in the next slide, where Pääbo was there. In fact, David Reich was the one who was leading this from Harvard Medical School. Pääbo and myself and some of our colleagues are part of the study. We have shown that these are the places where dots are there. These are the places the sample has been sequenced, of course, long back, 2016. And we found that we all carry some proportion of Neanderthal genome, except Africa you do not find, that is a 0. Then up to 3% of all of us are carrying the Neanderthal genome. There is also Denisovians, another human-like species. We also carry some proportion of Denisovan genome, up to 0.5%. And there are several papers again published by Svante Pääbo showing that there was an admixture between human and chimpanzee, also equal admixture between Neanderthal and Denisovan, okay, some time back. So, we will not go into detail. So, again coming back to this, that when did the modern human arrive? We talked about the early human evolution or evolution of hominids. But when we talk to the modern human means we are all modern human, right? We evolved in Africa about 160,000 years back. And the question is, from Africa, how did we migrate and populate all over the world? Right? That was a major question. But including geneticists, archaeologists, anthropologists, linguists, and very fascinating to see the they are the people with other expertise. You can see they are geologists, right? So, geologists also interested in understanding the human evolution, okay, even the common people. That is why I titled Who Are We? So, they showed in their studies, actually they dug out the place called Malawiya, where there is a lake, and that lake about 75,000 and between 135 and 75,000 years ago, there was a mega drought in the lake. Therefore, the water level has gone down. Because of that, probably the human who are living in Africa started moving out of Africa. That may be one of the reason. Then coming to migration of people from Africa, there are several hypotheses. Group of scientists including geneticists, anthropologists, archaeologists, historians, they believe the modern human might have taken the northern land route. Okay, there was a land connection, they migrated. But other group, they didn't believe on that because the climatic condition here might not have helped them to walk towards north. So, therefore, they believe that they might have migrated taking the southern coastal route. Okay. So, what is the evidence for that? Then the archaeological cited that there is evidence that modern human arrived in Australia about 50,000 years back, in China 39,000 years back, Middle East 47,000 years back, and Europe about 40,000 years back. But unfortunately, we do not have any archaeological site, very old site, to tell that when did the modern human might have arrived in Indian subcontinent. There are few sites. This is a site at this is a place called Jolapuram in Andhra Pradesh. The archaeologists have excavated the site and found only the stone tools, probably the modern human might have used to kill animals for their food. But there was no biological remain in that particular place. And there are a couple of places near Madurai, this is extreme south. One is Keeladi, other one is Achanallur. In Keeladi, two intact skeleton was found. Again, it was not very old, maybe around 2 to 3000 BC. In Achanallur, again, there was small broken bone, also not very old. There is one place where we could get analyzable ancient bone. It's a place called Rakigiri. You all know Harappa, right, where the modern civilization happened. There was an intact bone, the skeleton of a woman, where we sequenced and we found that bone also is not very old. That is only 3000 BCE. That consists of two different genomes. One is the Iranian hunter-gatherers, more than 10,000 year old. Other one is the Andamanis, which I will tell you in coming slides. Again, it is not telling about the early human migration into India's subcontinent. This is one of the biggest ancient DNA study. Ancient DNA from particularly Central Asia, we used modern DNA from South Asia. As you see, the large number of authors, most of the large scale study required good collaboration. Okay. You can see large number of authors again led by David Reich, and we are part of it. So, this again is not telling about when did the modern human come, but this tells that there was a group of people, steppe pastoralites from Yamnaya, they came through north and again not very old, 1000 to 2000 BCE, they brought language called Indo-European. And they also took the Indo-European language to Europe, not telling about early migration. In the absence of archaeological tools, we for the last 2.5 to 3 decades have been working on the contemporary population, those who are living, right, and we try to use their DNA and try to understand whether the DNA has any information where we can reconstruct the population history. While doing this, we surprisingly came to know the very first out of Africa human. I told you the human originated in Africa about 135,000 years back. And the very first modern human who migrated out of Africa is in Indian subcontinent. Anybody can guess who are they? Any guess? Sorry? Yeah, absolutely. The Andaman tribes, very close to Calcutta, right? So they are living in different part of the islands. There are 4 tribal groups who typically look like Africans. Jarawa, Great Andamanese, Sentinelese and Onge. 2 of them typically look like Southeast Asian like Nicobaris and Champans. While these 3, Jarawa, Great Andamanese and Onge, we have some contact and collected the samples, but Sentinelese are still in isolation. Nobody can go near them, they will shoot with arrow. It happened few years back, one American tourist tried to go there and they killed him. Again, you can see a lot of YouTube video explaining about the entire process. So we collected the samples from these 3 groups, Onge, Jarawa and Great Andamanese, but the numbers you see that they are very small in number. Initially we thought probably because they are small in number due to high practice of endogamy, they are marrying within the group, therefore not much of genetic diversity, therefore the number is still less. But we came to know that when the Britishers wanted to construct the cellular jail, this is a cellular jail, somebody visited already will realize, and those who wanted to go and visit, then you can see the cellular jail. Earlier there was 7 wings, now I think only 3 are existing. The way they constructed the cellular jail is that if each one is one cell. This is a front view and back view of other wing. They cannot see anything backside, they also cannot see except this. Okay, that is the way our freedom fighters have been kept. Okay. The reason why I am saying that for constructing the cellular jail, they killed some of the tribal groups. And I don't know what was the reason after killing them, they took their skull and kept in Duckworth Laboratory Museum by labeling like this, right. And they also took some the hair of them and the skull of them. You can imagine hundreds of years back what was the reason, but in fact that has helped us in collecting some samples. So we had a collaboration with Cambridge University, and there was an exchange program. Students from Cambridge came to my lab and my student went there. That gave the opportunity for them to collect some sample and analyze. So in addition to that, we went and collected the sample. This is Onge tribe, and in fact they are in a place called Dugong Creek, very remote place. We have to get the permission from government, central government, then the local government, then it's a very long process to go. We went from Hyderabad to Chennai, Chennai to Port Blair, from Port Blair to a place called Dugong Creek, not Dugong Creek, another place, by overnight ship, then from there by road, then by boat, right. And we were there actually for maybe couple of hours to collect. Then we have to come back to take the ship to return to Port Blair. So we collected the sample, try to understand about their origin. Okay. I hope you are all biology students, that if you want to do any genetic study, we have to look at the DNA. Where those DNAs are there is the nucleus. Actually, when the cell started dividing, then the nucleus become thread-like structure like chromosomes, right. So we have 46 chromosome in somatic cell. We inherit 23 from father, 23 from the mother. So that means that carry the information from both the parents. In addition to that, you know that this is a mitochondria, right? Mitochondria has its own DNA called as mitochondrial DNA. While we inherit nuclear genome equally from both the parents, we inherit mitochondria only from the mother. Okay? Mitochondria all know it is a powerhouse of the cell, right? So when I talk to students, I say that so all the power comes from the mother, right? ATP is generated by the mitochondria. So we inherit mitochondria only from the mother, mitochondria and mitochondrial DNA. So if we want to trace maternal lineage, we have to use the mitochondrial DNA. So we used the complete mitochondrial DNA. In fact, I will not go into detail, but you can just see the color of the map and the pie charts. These are mitochondrial haplogroup tree. It's a kind of evolutionary tree, parsimonious tree, we say that stepwise. Initially few mutation, additional mutation will put them here. If these mutations are not there, that may be different mutation that will put them here. Every time they accumulate mutation, they fall in different haplogroup. And you can see Africans have very old haplogroup L1, L2 and L3, right? Whereas you see that different continents have different set of mutation that we assign them in different haplogroup. When we sequence the complete mitochondrial DNA of the Andamanis, we expect, because they are typically like Africans, right? So they should have mutation which help us in putting them into L1, L2 or L3. But to our surprise, we found there are a lot more mutations. These are the site, mitochondrial DNA 16,500 base pair. In that stretch, there is a mutation at 200 position, there is a mutation at 1524th position. So like that, these mutations we could not find in any population all over the world. They are very unique set of mutation because they are in isolation for very, very long time. Similarly here, based on the mutation one can also calculate the age of the population. By doing that, we propose that the Andamanis are the first modern human who migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 years back. This is the first genetic evidence suggesting that the Andamanis are the first modern human who migrated out of Africa. In the same issue of science, there is another study by Richard Martin. They took a population called Orang Asli from Malaysia. So they also felt that the rapid coastal migration is the first route of migration, and in fact there was a perspective written by Peter Foster and his colleague citing both the study, our own study and Macular study, showing this map. So this is the first wave of migration. Okay, that remains today, there is no contradiction about that. After that we wanted to see the nuclear genome, the whole genome was sequenced, about 1 million genetic markers have been identified all over the genome, and we use that, and in fact we have predicted that the Indian populations have come from two founding populations. Who are they? One is ancestral South Indian, probably they are part of the early human migration. While Andamanis were migrating, taking southern coastal route, some group stayed in southern part of India, whom we call as ancestral South Indians. Then probably there was second wave of migration which happened through north, somewhere in the Middle East. One group has gone to Europe, other group has come to northern part of India, whom we call as ancestral North Indians. These are the two founding group of the Indian subcontinent. So why we say that they are more closer? There are several mutations which we found, I am not showing here, the skin color mutation, lactate persistence gene mutation, some of the haplogroups which are common in Europe also present in northern part of India, right. So, on the other hand, if you look at, this is very interesting to understand Indian population. These are all Indian populations. This is European sample, African sample and Chinese samples. You can see these three groups, although hundreds of individuals, they are in a single cluster. That means genetically there is no difference between the individuals, although it has been collected from different part of Europe, different part of Africa, different part of China. But look at Indians, every single population is unique, uniquely clustered. But another interesting aspect is, although every single population is unique, but we all have some kind of genetic link, right. That tells that India is not inhabited by a single large population as we see here, but India is inhabited by large number of small population groups, but we are different, but still we have some kind of genetic affinities, unity in diversity. And what we said at the time is that all Indians have admixed. Because of this, we refuted Aryan invasion theory. This is a theory proposed by Max Muller, that the white skin colored people are Aryan, they came to northern part of India, they pushed all the Dravidians who have dark skin color, but we did not see any distinct population in India whom we call it as Aryan. We also propose that there is a high frequency of recessive disease, which I will come bit later. But later what we have found was these two groups, because of this there was an admixture, right, between ancestral North Indian and ancestral South Indian. That is the reason there is a kind of gradient of clusters, whereas these groups are very distinct. But when this admixture took place, we wanted to estimate, we took large number of samples from southern part of India, large number of samples from northern part of India, and we have shown that initially these two groups, ancestral South Indians and ancestral North Indians, gave rise to many population independently. You can just imagine the first group settled in somewhere in jungle, we are talking about 65,000 years back, right, and they must have, some group must have finished the food which was available there, and they wanted to go to a better place, okay. Some group have migrated there and from there to another place. So now after migration, these two groups are now very distinct, there is no mixture between these two, and they again practice endogamy, means marrying within the group, right. So that makes every single population, because they must have acquired unique mutation which makes them very different from each other. So that is how both the founding groups ASI, that is ancestral South Indian and ancestral North Indians, have happened. But during the last 2 to 4,000 years, these two groups have admixed, okay. You can see just color indicates the genome is admixed, genetically admixed. But after admixing, every single population started maintaining the endogamy. Endogamy means marrying within the group, within the caste or within the tribe. So these are the three aspects for the human population history in India. Initially there was a migration, later migration, these two groups have independently gave rise to many population. Last 2 to 4,000 years there was an admixture, then last 2,000 years every population is maintaining the endogamy, means marrying within the group. Now we have to see what is the impact of marrying within the community, either it's a caste, either tribe or any other group. So that is what I am going to take you through. So in order to understand that, we took another study. So this time we are not restricting to only India. We wanted to expand it to other South Asian countries, including Sri Lanka, Pakistan, Nepal, Bangladesh and so on. In this study we took 275 population and we collected 2,800 individuals. The reason for why we took other South Asian country is that in our earlier study, we found there are some mutations which cause cardiomyopathy is very highly prevalent in South Asia, but not in other countries. Therefore, it is important whatever we see in India is also true for other South Asian countries. So, in this study, we actually come up with idea that there is a lot of opportunity for mapping population specific recessive mutation which might cause population specific recessive disease. I will tell you the coming slides. More technically, it is called as IBD, identity by descent score, but again I will not take you in detail, but just simply look at the line. Each line represents one population. Okay. If the IBD score is more, that means in a particular population they share more common haplogroup or haplotype. Okay. That means if more relative, if you take two unrelated individuals and compare their DNA, maybe some 10 kb region, if they are related, you do not see any variation, but if they are unrelated, you will see variation at every 500 base pair or 1 kb region. So, based on that it is calculated, if the length of the IBD is more, that means number of individuals whom we analyze, they are more closely related. Okay. So, what is the impact of that? You can now see these two populations, Ashkenazi Jewish and Finnish population. Their IBD score is too tiny, right, but still they have large number of diseases. These are the diseases, these are the genes, but most of them are autosomal recessive. Similarly, Finnish population. You can broadly classify the disease into two categories, of course X-linked, Y-linked also there, but broadly autosomal dominant and autosomal recessive. I mentioned that we have 46 chromosomes, 23 chromosomes we inherit from father, 23 from the mother, right. So, that means every chromosome we have 2 chromosomes. If you take chromosome 1, there are 2 chromosomes or 1. One we receive from father, one from the mother. If one of this pair has a mutation, if it leads to the disease, it is a dominant. One of the pairs has a mutation, right, it is a dominant. But if it is the recessive, if the person has a mutation in one of the pairs, it will not show any disease. So, dominant disease one can see easily, because somebody has a mutation in one of the pairs is going to have the disease. But the problem with recessive mutation is that they will remain as a carrier with one mutation until otherwise somebody has mutation on both the chromosomes of the pair. So, that is very, very challenging in India. So, you can see the recessive mutation. So, we predicted that at least 1/3 of the Indian populations are expected to have population specific recessive mutation. For the students, I will just give you that how one can imagine that how important to study each population who are predicted to have carrying the recessive mutation. This is pedigree. I think most of you are aware, the biology students, right. Circle indicates female, square indicate male. And if you put line, that means these two are married, one line if you put, and these two are their offspring. That means in this family, the mother carrying the mutation in a heterozygous condition, because it is a recessive mutation, she is normal. She married somebody who does not have the mutation, right, and the next generation she is transmitting that to both of her daughters, right, and there again in heterozygous condition no problem at all. So, she married somebody outside, she also married somebody outside who does not have the mutation. Until here, generation after generation it is going in a heterozygous condition. But look at here, these two individuals, now they say they are unrelated, okay, but they have come from a common ancestor, but genetically they are related, right. If these two marry, you can statistically say that there is a 25% of chance that she contributes this abnormal chromosome, he also contributes this abnormal chromosome, therefore there is a disease, recessive disease. So that is very, very common in India. I will tell you a couple of examples. There are two pedigrees. This is consanguineous family marrying within their family. This is a non-consanguineous family. You can see the impact of the mutation, just single base out of 3 billion base pair, what kind of effect it would give. You can see this is a gene called polymerase gamma. The gene is essential for mitochondrial DNA replication. You all must have studied the replication, right. So there is a single base mutation. This is a wild type, this is a complete mutant, this is heterozygous. That means 2 chromosome, pair of chromosome, right. So if the sequence has 2 different chromosomes, one chromosome carrying the wild type like C, what you see, other one carrying the mutant. That is why it is called as heterozygous here. Both the parents are heterozygous, they are absolutely fine. But look at the next generation, 2 out of 3 are unfortunately have the homozygous mutation. Father contributed the mutant chromosome, mother also contributed. Therefore, the children had encephalopathy, seizures, stroke-like episode, and most importantly premature death. They died at very, very early age, even less than few years, 18 months, 9 months and so on. So such a devastating effect of a single base mutation, it happens because of the consanguinity or the endogamy. It also causes brain lesions and so on. Now coming back to this slide, because in this slide we predicted that there is a large number of population in India expected to have a population specific mutation might cause population specific disease. So we wanted to confirm that. We took few families, few population including the population called color and few more, and we sequenced. And we found, actually we went back to the population, there is a disease called junctional hereditary epidermolysis bullosa, the body full of rashes and congenital heart disease. The children with the mutation 11 base pair deletion in a gene called LAMB3, they die at very, very early age, less than 2 months. There is also internal bleeding and so on. In this case again parents are absolutely fine, normal with heterozygous mutation, and you can see the pedigree again, parents are normal with the carrying the mutation, but the children, 2 of them have died. So we wanted to actually look at the frequency of that mutation. I think something is missing here, maybe the same image. We looked at different populations from India. We also looked at different databases, and we found only 2 South Asians carrying the mutation heterozygous condition, but we looked at 8,62,000 individuals. When you look at the chromosome, it is a double, right. 17 lakhs worldwide genome does not have the mutation except 2 individuals. That means this mutation is very, very specific to that particular population. There are several such populations are existing. Until otherwise we go to a particular population, analyze their genome, we cannot actually predict. So this is very, very common in that particular population. There are 4% of the individuals who are carrying that mutation. Now this is again interesting. If the mutation is novel, it is not found so far, and we have to see biologically how it has the impact. If you look at the wild type, it produces 1172 amino acid, because these are important for all the functions. So, in this case, when there is 11 base per deletion, it actually truncates the protein, 799 amino acid only. So, the remaining amino acids are not there, therefore, this protein cannot do its function properly. Any new things, we have to generate the animal model and see whether we see the same phenotype in the animal or not. When we introduce the mutation using CRISPR-Cas, there is a new technology which one can do the gene editing, and created the animal model, you can see that animal also has the same impact as what we have seen. Even the internal organs, you can see that there is a internal bleeding and also inflammation. We are further looking at it in a at molecular level, that what actually it leads to. So, this is one aspect. The second aspect is, as a responsible scientist of this country, it is important to help the family whatever the best we can do it. They are in very, very remote place near Madurai. We wanted to help the population. There are 2 mothers who are pregnant, where there was a children who have died because of this genetic issue. Now, we wanted to see whether the fetus carrying the mutation in a homozygous condition, if that is the case, the fetus again it will grow like the way which I showed, and again there will be a disease burden in the family. So, we wanted to avoid that. We made a simple PCR based method to see whether the fetus carrying mutation in heterozygous condition, that means one chromosome carrying the mutation, other one is normal, or homozygous mutation. So, we found this is amniotic fluid, and this is the homozygous mutation, this is the heterozygous, and you can see 2 bands suggesting that it is heterozygous condition. That means one chromosome carrying the mutation, other chromosome is normal. Therefore, we advise the mother to continue with the pregnancy, and it is our joy to see that the child born last June absolutely normal phenotypes, but carrying the heterozygous mutation. So, that gives us much more joy than just doing our work in the laboratory. So, you can imagine that how studying the population history would help us in understanding the health condition of the population. So, now wanted to screen the entire population. To some extent, we had a camp and collected the sample. Now, we are approaching the government to give us a permission to screen all the populations. So, in that context, I wanted to show towards the end of my talk that in CDFD, Center for DNA Fingerprinting Diagnostic, where I was heading that institute, where your director also was there earlier, and we had a mission program on pediatric rare disease. So, exclusively identify the unique phenotype, sequence them, predict the impact of the mutation. Then we wanted to create different animal model, which I showed earlier, whether the mutation is real cause for the disease or not. Then try to treat them using gene editing method, and in that way we can help in family not only prenatal diagnosis, actually we can also give premarital counseling, and the boy and girl who are marrying, who may look normal today, but they may carry the mutation heterozygous condition, whether they carry heterozygous condition or homozygous, not homozygous, or there is no mutation at all. Based on that, we can give the counseling whether they can go ahead with the marriage or not. So, that helps in removing the disease burden in the future. So, that program was launched last year by Dr. Rajesh Gokhale, the Secretary of Department of Biotechnology, Government of India. I think it's going very well. So, with that I would like to thank those who have contributed this work, my colleagues and collaborators. As I mentioned earlier, collaboration is very, very important, and you can see the list goes. The funding has come from CSIR, UK India Education Research Initiative grant, Department of Biotechnology, Department of Science and Technology, and Medical Research Council at London. This is one of the ongoing projects. Thank you very much. Any question? I'll be happy to answer. Yes. Yeah, 1 minute.
Thank you, Dr. Kumar Swamy sir. Actually, I was very happy first time when you are studying this speech. I was very interesting this, but last part is really painful for me because my son was first child was died for these mutations. I also go for Nivans for 2 months and staying there. Dr. Modhu ma'am, another Dr. Modhu ma'am. You are talking these genetic mutations. Actually, my child in the year of 2015 was born, and 2018, as of 3 years, he died. This is a type of congenital delayed development and seizures, and this is a COLA A6 this genetic sequence is different, and I have tested several tests. NEP, you know that next generation NGS, next generation sequence test, which is done by the MedGenome Lab in Bangalore. There is no any. We have also done our genotype and phenotype test sequences, but there is no any difference in I and with my wife's. But they did not diagnose till now the disease. What disease the name? This is my test is here. This is uncertain significance.
Okay.
Ullrich dystrophy type. They suggest.
Okay.
They told that this type of mutation maybe this type of mutation maybe the 25 or 26 genetic years back they may have. I know that just like example for the me, this is a white rose plants, but after 10 years there are some pinkish flowers. That may be happen in your child, but you told that after next, you have also go. Can I take a next baby after when when we are planning for the next baby, this suggests me there is no any chance, and of 99% of 99.99 you can take a child. And fortunately, thanks to God, I have got a lady. I have got a baby girl. She is 4 years old, doing a very well, going to school.
Very good.
I'm very happy. This is usually occur here. We have no any. We have no any. Actually, we treat my baby as the last year. We want to go to America, USA, because my relatives are there. My uncle is the Kentucky University research principal scientist. They also suggest me to go there.
Right.
But here the doctors told me that if you go to the USA after changing all the genetic sequence, there is no any definite confirmation for me if the baby is maybe cured or not. So I decided to stay here, and she also told me decide to stay here. What will happen? They also told me that your baby is no longer tomorrow, maybe 1 2 months or 3 months. After 3 months, she died. He died. So this maybe happen. Don't worry. It is sir. Can you tell me how can you prevent this implications of for mutations to?
No,
Actually, we are the unknown couple. We don't know. She also told that you have no any symptoms.
Right.
This may be happened. This may be happened.
Okay, sometime the mutation is de novo. It happens in the germ cell, either sperm or ovary, in the egg. So that you may not see either in yours or in your wife blood DNA, right? That can happen also. But mostly the inherited disease, parents would carry that and transmit to the next generation. Particularly in the recessive mutations, who carry the mutation one of the chromosome will absolutely fine, right? But this is one example. There are many examples which we have. I could not show because of the limitation of time. So that is very, very high in South Asia, not only in India, because of the family structure of the population who marry within the group. But thanks to the genetic diagnostic method, that we can still help the family which has those mutations.
Nivens also takes my baby's brain samples and also the muscles
Tissue,
of,
yeah.
for further any research. They also take my emails and further any development grow. He
Right.
also inform me.
Yeah.
Sir,
They
there
have
is another question. Why the first humans, early humans come in South Africa? You told me it may be the Amazon, maybe the lands, the wetlands. The cause of this, it may be the wetlands, the woodlands, the food area, the grassland, Amazon jungle, rainforest. Maybe the one of the reason.
No, the origin itself is of Africa, because that's where there are many hominid species. They all evolved there. And then started migrating out of Africa, particularly human, when other species have vanished. There is no other place, although multi-regional origin has been proposed, but there is no evidence for that, genetic evidence.
Thank you, sir. This is a very good, interesting and awareness talk. Thank you, sir, again.
Thank you.
Sir, so my question was that during the first half of the presentation, we saw the map of India, and then there was a divide that the ancestral North Indians and the ancestral South Indians. So my question is this coming to the conclusion that both of them have a different genetic history, both come from different lines, can't it be so that they both of them came from the same land, they came from the same line, and then after that both of them differentiated because of geographic reasons? And this is the one doubt. And the second doubt I had was that initially you said that they might have intermixed, and that is why we see the convergent thing. So my question is, if they were doing the normal mating, random mating before, so what could have been the behavioral societal pattern due to which they started doing the endogamous, they started marrying within their communities or maybe within the same regional. Yeah. Why did they do this linear, why did they marry within the community later on, but initially they were freely interbreeding?
Yeah. I think actually you're asking again to summarize my talk, right? Yeah, please sit down. So I mentioned that everybody, if you trace their origin, it will end up in Africa, right? So there's two wave of migration. One came to southern part of India, particularly the Andamanis, who typically again look like Africans, right? So they stayed there for the last 65,000 years without any admixture, right? On the other hand, those who migrated towards north, they accumulated different kind of mutation where the phenotype has changed, the skin color mutation, for example. Because to live in a condition where the sunlight is not always exist, but the sunlight is essential for vitamin D synthesis, right? Therefore, there was a mutation in the gene called SLC25A, which turned the skin color into lighter, therefore the melanin content was less, and those who had probably survived better because of the ability to synthesize vitamin D. So that became common factor in that area. Those who do not have the mutation must have gone, right? It is selected for that reason. And those who are carrying the mutation must have come to northern part of India, right? Then there is admixture. You can actually see if you take extreme south and extreme north, you can see how much of the ancestral South Indian and ancestral North Indian genome is mixed with them. If you go to south, they may have 80 to 90% of ancestral South Indians, and there are some population who are closer to Andamanis. There is a population called Kurumba in Kerala. They are more closer because they are also in isolation in a forest, right? So then you see that 10% to 20% of ancestral North Indian genome. On the other hand, if you go to extreme north, they may have 90% of ancestral North Indian and 10% of ancestral South Indian. The color itself actually gives us a kind of indication that how much of the admixture took place. Unlike other countries, if you go to China or Europe, they are all with white skin color, you cannot differentiate. But in India, we have extreme light color and extreme dark color and in between so much color. That itself says that we are all admixed. And the last point you asked that why the endogamy is being practiced.
Later on.
That is our social structure where the caste system which came around 1000 years back or before that, based on the job they do, probably put more pressure. So those who have specific job, they marry within the group, and the caste system came again marrying within the group. So that makes every population as unique, because we are practicing endogamy. Whatever the mutation happens in one group, that remains there in that group. That is what genetically differentiate from the other group.
Sorry, really we have to stop. We have another talk after this. I'm extremely sorry. Firstly, let's thank Dr. Thangaraj once again for a very engaging talk. So please gather people at lunchtime or any other time and ask any questions. That is most welcome. So thank you.
Thank you, Professor Thangaraj. It was such an honor to have you among us today. We have our next honorable guest, Professor Oyun Banerjee, Professor and Chair Member of Board of Directors, Rice Foundation, ISER, Kolkata, among us now. May I request Sir to kindly come up on the dais, please, and Professor Ghosh to kindly accompany. I would request Anushmita and Payal to kindly felicitate Sir with the bouquet and the replica Raman spectrometer. Professor Ghosh would be introducing Sir to us. Ma'am, please.
Okay, so I'm going to keep it as short as possible in the interest of time. Oyun Banerjee, or Oyunda as we call him, is presently a professor and dean of students, and he's also the chair of the Department of Physical Sciences at ISER, Kolkata. He is also one of the founding directors of the Research, Innovation and Scientific Entrepreneurship, or RISE Foundation in ISER, which is a Section 8 company formed inside the premises of ISER, Kolkata to incubate deep science-based startups. Professor Oyun Banerjee, he did his PhD in physics from IACS Bangalore and joined General Electric Global Research in Bangalore as research scientist. Since 2009, he has been in the Department of Physics at ISER, Kolkata. So with that, let me ask you to take over. Yes, there's a caller mic. Yeah, you have.
Okay, thanks, Devusri, Dr. Ghosh. We call her by another name. I'm not sure she'll be very happy if that be announced anyway, so maybe I'll keep that a bit secret. But I know her for the last, how many years? 20 years? 20 years. I know her for the last 20 years. That is remarkable. I mean, I can't believe how old we've all become. So, yeah, so you heard a lot of things said about me, but the capacity in which I'm going to speak today here is how I reached here to some extent, and also the fact that about 25 to 30 years from now, before now, I was sitting in that place, right? I was sitting there, and of course those days I do not think, we had not even heard of something called Science Day. I will blatantly admit that, and I had never thought that time, that 25 years from now, I would be speaking on Science Day, coming from 80 kilometers from here on Devusri's invitation and coming and talking to you guys. So that when you just think of it, it appears a bit strange, because I studied in a school, a very, very famous school in Kolkata. It's in Park Circus. And about 200 of us appeared in ISC. It was a ICSE board school, ISC school. 200 of us appeared in the class 12 board exams. I can tell you, to the best of my knowledge, and I think it's quite definitive because we have a big WhatsApp group where almost everybody is included. There were 2 scientists, two out of 200. And I said were because the other person than me was here. And it pains me unbelievably to say was, because Professor Bhushan Majumdar somehow tricked us all and, escaped the clutches of Maya some time back. 2 out of 200. But I can also tell you, the rest, most of the rest of 198, they are very, very successful. Most of them are very successful. They are doing good things and they are very rich, they are happy. But it's unlikely that they actually come up on stage and speak to youngsters. It's a bit unlikely. They are businessmen, they are all kinds of things. Men, because I was in a non-co-ed school, boys' school. And actually some of them call me and tell me, they are very rich, they are CEOs of companies and they have their own businesses and all kinds of stuff. And they tell me that, grudgingly, a bit grudgingly, that, see, what will you do tomorrow? So I said, what question is that? So he said, no, what is the plan tomorrow? I said, plan, I'll get up, I'll go to ISER, I'll go to my lab. I'm sure something will happen which is really strange, which will drive me crazy. And then maybe I'll do something, but something new. Surely something which I did not expect. And that is where they say, good, nice, I will also go. What they don't tell me is that they'll also go to their office and do all things. But it is unlikely that they would do something which they did not absolutely expect the previous day. And that is the difference. And that is the difference of being a scientist, of being somebody who is really creative. Of course, this is important nowadays. Money is very important, but this is getting very, very crucial these days. Does money get happiness? I'm not going to get philosophical and all, but the fact is, that is really a question. And nowadays, of course, if you do science, then it is not that, I remember when we were actually getting out of school, very, very, very few students of friends of ours wanted to get into general sciences, because I remember one guy who very, loudly said, "Are you crazy? Physics? Physics? Do you think I'm going to sell egg roll in the footpath?" Precisely this is what he had said. So here, not selling egg rolls. Though I like egg rolls, but I eat them, don't sell them, right? So this is what we do for a living. Our lab is called Light Matter Lab, and because we look at interactions of light with matter, a very small matter, very, very small, 1 micron, 2 micron particles, not nanoparticles. We are not in the quantum domain, but we are still at very, very small length scales. What is very interesting is we can actually make these particles dance to the tune of light. As you can see, this particle is nicely revolving and rotating like. It is almost like the Earth in the solar system. You see all these particles are arranged in a nice necklace. They are actually the necklace is becoming small and big. We can make particles dance. We can also make them sing. You know, I'm not joking, really make them sing. And I have heard them sing. About 12 years back, when we first set up the lab, I had one of, you know, the first batch of BSMS students of IACS Kolkata. And they are the ones who set up my lab, the two of them. One of them is a faculty at SN Bose now. The other has a company in Singapore, multinational company. So what he did, so these particles are trapped. I'm not going to go into that kind of details because whatever I'm going to say here about, I am not going to be too technical today. This is not the forum. I want, if of these maybe what, 150, 100, whatever number, if one of you is talking here after 20, 25 years, I will consider my time well spent, if one of you. Anyways, to come back. So these particles, after they get, so I can trap them or confine them. So these are actually inside water. And I have a microscope and I focus light very strongly. So you have seen a magnifying glass, right? When you have sunlight falling on magnifying glass, through a magnifying glass, then it burns the paper. Why? Because the light focuses, the light is focused by the magnifying glass to a very, very small spot. And there is something called intensity, power by area. The intensity is very large because the area is very small. So the paper starts burning. So we actually have something like that. We have a very, very small focal length objective lens in a microscope. So how many have seen a microscope? Raise show of hands. You know there's an objective lens, right? Magnifying 100x, 1000x, you've heard, right? You know that? So we have this objective lens, 100x, focuses light to a very small spot, and that light can actually trap particles, confined particles, which respond to light. What happens is when you trap these particles, they are not still, they actually jiggle around. They perform Brownian motion. That Brownian motion, you've heard of Brownian motion, right? This Brownian motion actually is like noise. It has all possible frequencies embedded in the motion. Okay? So it basically moves at all possible frequencies because it's like noise. What we had done is that you can actually study, you can measure that Brownian motion by putting light again from a different laser on that dancing particle. Now when the particle moves, the light is scattered back of it, right? And the light intensity changes, and it changes at the same frequency as the motion of the particle. Now if you feed it to a detector, the detector actually gives you voltage output at different frequencies. And that is what my student had done. So I had actually a small radio in my lab. So it was my radio because again we were trying to do something cute, because we were trying to extract a certain part out of the radio because we did not have that part. It was a detector, quadrant detector. And we had to extract it out because we could not buy one, not because we did not have money, but because our office had lost the purchase order. Okay, so we were there, we were having particles, we could not measure anything because you need these specialized detectors. And would you believe it, CD players had that. So what my students did, they went to that time this place at ISER. So do you know where ISER is? How many have heard of ISER? Oh my bad, that is really bad. We are disappointing. Huh? Close to Kalyani, Mohanpur. Very good. AIMS. So now everybody knows AIMS. Because of AIMS, I think we are getting famous. Anyway, so that time, now if you go there, it's a pretty developed place actually, pretty developed. That time it was a panchayat. So there was one radio shop, electronics ka dukan, and the student went there. He got these defective CD heads. This guy thought we were completely nuts, because why would you want these CD laser, these heads, optical heads, which are defective. The reason was that when the CD heads get defective, the laser blows, the CD doesn't blow. So what he did was, we plucked out the detector and we were using it. So what you do is, now if you put that input, this scattered light into the detector, it gives out a voltage. And that if you connect, so my radio was still working, the amplifiers were working, the speaker was working. If you connect that to the speaker, then the speaker gives you sound. And that sound frequency changes depending on how my particle is moving. If my particle is jiggling very fast, high frequency, it will go like, "Oh." If it is slowing down, it will go like, so have you heard, so when we were small, when we were very young, we didn't have phone to chhodi do, we had TV which used to run for 2 hours a day, 3 hours a day, 6:30 to 9:30, 3 hours. So our life, our entertainment was the radio. So has anybody tuned a radio and heard static? Static, yeah, that we could produce in our lab by trapping an optical bead, trapping a small, we call these beads, particles by light, and my student called it bead radio. We didn't publish it, we thought it was too trivial. 2 years later, there was a PRL published basically on that, close to that. Anyway, we have a lot of things you can do with these kind of nice, because these particles, once you trap them, you can study them for a long time. Very, very useful, because you can trap them for hours, and they give you all kinds of beautiful information about something, about the property of light itself, about statistical mechanics, all some very, very nice things. I'm going to talk to you a little bit today about another thing we discovered, and that is using bubbles. Very different. So we can actually write using bubbles. Anyway, that comes later. My journey, a little bit, I have to tell you, I was never a star student ever. Once in my life I came first, that was in class 2. Uske baad never. Class 9 I was third, because the good student who used to come in first, second, uska chicken pox ho gaya, he could not give the exam. So I came third. Okay? This was crucial, because everybody, all males in my family were engineers, and I promised I would not be, whatever happens, I will not be an engineer. And my father was very disappointed to an extent that I flunked joint, IIT, everything I flunked, thoda sa willingly, but I was not great also, right? So even in BSc, when I got decent result, even then he was, hey, radio physics, any chance? Radio physics ka form tick, BTech radio physics was very good in Calcutta University those days. Can you do? No. Strictly I did MSc, no engineering. Whatever it is, I will not do engineering. And this was important too. I wanted to do something creative. I was involved in theatre, group theatre even then, now too. Our friend Devastri knows that well. I don't know whether you guys, I mean, okay, yeah, she is herself has her parts there as well. But I just follow, since I did not do engineering, and since I did not have the guts to do creative arts, I wanted to go to a film institute, but no way you can do that. At least you could do that in a Bengali middle class family in those days. Okay? So I had to pursue a formal course of education, and that was BSc, MSc, PhD. I didn't do a postdoc, but I have never got out of India. I've always been in India. But the interesting thing, I do many things now, but the only the thing that I've been taught formally is physics. And I really think that without a formal education, you probably can still, these days so sure, you can be a singer, you can be a dancer, you can be an actor, you do one TV serial, you get lots of money without any training at all. But physics and sciences, you need training. But what did the training do? What did the training finally do? It taught me to think. It taught me to think, and that is the most critical part. Any education that you have, unless it teaches you to think, it is worthless. I have my, this lot of friends that I have from my school, many of them I see they have done, maybe in higher degrees, but they still seem to be educated by WhatsApp University. They still seem to be having no opinions of their own. They just follow whatever is being told to them. The type of education, unfortunately, we impart in India still does not actually empower you to think. And that is what this pursuit of science can actually help you in. So I did not go into a postdoc because personal reasons I had to stay in India. I joined a company, I did research. And that was very useful because academic research and corporate research is very, very, very different. That's what I realized. Because, you know, in academia, you work for papers. One paper is your life. Each paper is like a drop of blood. Corporate doesn't care about papers. They want products. But in corporate, I could actually understand what is something that excites the world around me. That was very, very crucial. That 5 years. But then it also told me that I was straitjacketed. It was like that. I could not just get out. I couldn't do what I wanted to do. I left corporate. Thankfully, those days the ISAs were just starting. ISA Kolkata, by the way, is the oldest ISA by 2 days. Then it's older by 2 days than ISA Pune. And that time, ISA Kolkata wanted to develop an optics lab. I have developed labs all my life. I joined at ISC. My advisor had joined 6 months back. I joined at GE. There was a room they showed me, nothing, just 4 walls, empty space. Lab karna hai. I did. I just continued that in ISA. And the journey goes on. Okay? Now, as I said, why do I choose science? You know what? The other day, I was having, I was in a restaurant with a friend. And that friend is not a scientist. He's one of, not one, he's probably Kolkata's most famous light designer. He's mostly in theatre, but he's also now graduated upwards. He does light scheming for school halls and all kinds of things all over India. But he's a light guy. But he's very, very different from the light I do. And we are good friends. So we're chatting, and then he said that he wanted to know the nature of light, the physics of light. So I actually showed him the video I showed you in the first slide. The particle is revolving and rotating. He was like, wow, what is this, what and all. So I was trying to explain. While I was doing it, I suddenly realized that at that moment of time, no one in Kolkata is a city of around, 2 crores people? No one, not a single person, I can say this with confidence and with certainty, knew about what I was showing. Not a single person in Kolkata at that point of time had any idea of what I was showing this guy. In the whole country, I know there are about 7 to 8 people. 2 of them are in my lab. One of my main collaborators, he was at Hyderabad then. The other one was actually also in Hyderabad, 2 of them in Hyderabad, and 2 students of my collaborator. These are the people in India who actually understand this. But in Kolkata, at that point of time, not even one person knows this. That feeling is empowering, guys. Empowering. I may not have money. I will never ever in my life have the pleasure of owning a Mercedes or whatever. Not even Mercedes, the best cars in India. I'm not good in cars. Whatever, I will never have that, I'm sure. But this thing that I have found something which nobody else knows. Money never can buy that. I can tell you. Rewards, I don't need to stress. Material actually is there because I can tell you the time I had joined ISC, you know how much my advisor, we used to get 2,500 rupees. That was the JRF scale that time. My advisor used to probably get, because he used to stay in campus, so he did not get HRA. He probably got 1,000 rupees more. It was really bad those days. It was probably, if you sold egg rolls in the street, it was probably better in terms of money. It has completely changed nowadays. Nowadays it has changed. After the 6th Pay Commission, I can tell you, at the moment what it stands, actually a full professor in one of these national institutes of India has greater purchasing power than a full professor at Harvard or Princeton. That is a fact. Purchasing power in India, I mean. Not in the US, of course. US mein to bhik mangwa, wo chodo. But if you are actually in India, the money what money can buy in India with your salary is actually more than that guy can afford in the US. So it is not bad. It is not bad. Not even that, even if you're a school teacher at KV, it's also not bad. Salary is pretty good. Salaries are good now, believe me. Of course, engineering, right? I have a relative, he's much younger to me from my wife's side. He cracked joint. Well, he did not get the top Jadavpur and all that, but pretty good. One of these just lower rank colleges, just after Jadavpur, private college, but a pretty famous one. He did quite well. Believe me, the job he got after that was 8,000 rupees. 8,000 rupees is he had 12-hour duty. 8,000 he graduated to 12,000. Uske 20,000. Finally, after 12 or some like that, a dozen years or so, he's got some ground below his feet. He's in Bangalore now, he's better. Then what is so great about engineering? If you do a PhD, you get 37,000 now. And uske upar you get free travel to conferences, even abroad. Which company will give you when you get in? You think engineers, when they go to the US and all that, when they go for traveling outside, you know what the situation is there? I have known people who have been in the US for 2 years and they have not gone anywhere else except the county they are in. Because they work 6 days a week, 15, 14 hours a day, Sunday is just sleeping. That is called a life. That is called money. What will you do with that money? By 40, what is your, every day is a carbon copy of the previous day. IIT professors, I can tell you, there is a lot of angst because everybody wants to do software. So then why civil, why mechanical, why electrical, why architecture? Finally, if 90% of people who get there, who get out, with the degrees, finally go to jobs in software, what is the purpose of it? That's a question that is actually plaguing everybody here in this country, right? And I can also tell you, I have been external examiner in IIT Kharagpur several times. I mean, IITs are good, much better, but the private colleges, the people who teach, I mean, it's as if you don't care about any education. Only thing is, after 4 years, you get a degree, do a management degree or something, get a job. It's like a buzzing word in your ears. You get a job and then what? Yeah, you have 50, 60, 70 years to live. Is this what you want to do? Get a job, wake up in the morning, do some net surfing, nowadays nobody goes to the bazaar and this thing, everything comes online. Wo bhi kaam kam ho gaya. Then you go to office, do something, come back, see TV, then maybe go to a party if you're lucky enough, then come back, have some, if you're in that line, maybe have a whatever, peg or two, and then go to bed, finished. Next day is the same thing. Where is the creative, I mean, what is going to drive you? Then you have a heart attack one day and die. What is the legacy? I mean, what did you leave behind? This 35, 40. So much strain, so much stress, and to do what? Can they, I don't think there are too many engineers who will be able to say that there is something I know which 2 crore Calcuttans don't know. Very unlikely. I'm not demeaning anything. You can be, see, the whole thing depends on whether you want to do something in your life where every day will not be a carbon copy of the previous. And that only happens when you know how to think. And that, I can tell you, a science education can actually give you. Because the science for me is a way of doing things. It's a way of thinking. So if you're in a class, and how do you learn to think? Because, and believe me, it is an art, it's a process. It is not easy. I realize now that I had learned nothing, believe me, nothing, probably till my PhD. I knew some things, some techniques and how things work and some problem solving. Problem solving माने books में जो है उसका solve solutions और कुछ नहीं. In fact, when I went into my thesis, my lab at IACS, my friend joined. I mean, two people joined. One from me and the other my batchmate from Pune. That guy's father used to own a radio store. Radio TV repair shop. And from his birth, probably he has been soldering. Now, I come from Kolkata University and Jadavpur University. I had never seen a solder in my life. And there was my advisor, who actually, I remember in my interview at IACS had asked me, have you taken apart a cycle? I did not even know how to ride a cycle those days. Because I was in Kolkata, right? You didn't need a cycle in Kolkata. It's like your parents thought they will get run over very easily. All my life, I've lived in the busy streets in Kolkata, amidst the busy streets of Calcutta. And I can tell you, I was a complete disappointment to my advisor, because here I was a complete, theory guy who had never done real things never used his hands. He used to continuously ask me, why are you so bad with your hands? And has to sheepishly say that, nobody taught me. And that was the fact. And this guy, of course in my quantum field theory exam, he completely copied from me. But he had grown up repairing TVs. And he was like that in electricity. He's still one of the best experimentalists in the country. Fantastic experimentalist. He had a knack. But the exposure was great. Right? So the important part in science that I feel that science is finally a method of understanding things. And for that you have to be interested in how things work. You have to read, not textbooks. Wiki, so much of wealth of information is around nowadays. So much. You feel interested, just do a wiki. And then if you're really, into it, wiki gives you many references. Go on. And then ask questions. Ask questions. No question is stupid. You cannot believe, guys, I am teaching this second year in ISER, second year lab course, the optics part. That lab I set up in 2010, 14 years almost gone. This semester I have learned 3-4 new things. That is unbelievable. This semester, and everything the students have, through the students. They have asked me some questions that, "Bhai, ye kya hai?" Then, aisa hota hai. They have some, one girl, she turned her torch open, her mobile torch open and just put that in front of the grating, diffraction grating. And I was, bhai, ye to main kabhi nahi kiya. And we learned new things. And just that, in fact, this is one profession. If you're a teacher, you can never get old. Never get old, because you're always surrounded by young people, young, bright people who ask you, challenge you. School teaching, that's one of the most neglected things in the country. And yet that is where the greatest issue is. Anyway, that's for a different thing. So, physics. I am going to talk about physics because I'm a physicist. So the work that I'll tell you about, that has many things other than physics. It's like science, everything. So this is one subject where actually you have a class of theoreticians and experimentalists, and they are very different. Chemistry also has to a certain extent, but not in the way of physics. Physics is you have theorists, you have experimentalists, two different classes. And that is useful because the skill sets are different, because they are very complementary. And what do you have in physics? I'm sure you've probably heard of this. All these, you have condensed matter, you have optics, you have soft matter, astronomy, high energy, statistical, lots of things. And believe me, there are lots of institutes now. Presently, IISER Kolkata is hiring. We are having interviews of talks basically of applicants who have done their mostly postdocs abroad. And we are seeing many of them are very good, but there are also many who are good, who have good papers, but that thinking part, there is still a problem. And we actually want to take people. Believe me, there are many jobs now, and there aren't too many competent people. There aren't too many competent people, right? You have many, so TFRs, these are theirs. So I can tell you, this is also, of course, nuclear physics. My boss used to say unclear physics for him. So jobs-wise, you have now 23 IITs. And I can tell you each IIT has some, it's a big, Bombay IIT, can you believe? I was stunned when I went there in December. That population of the campus is 20,000. That is stunning, right? 20,000 people. Faculty is about 600, 700. Remarkable, right? You have now NITs, you have these central universities, state universities, private universities. DRDO. You can read, I'm not going to talk about this, but the final thing that I'm going to tell you is that it is not that science cannot give you jobs. Science, it is not that even in DRDO, finally you are in research. Finally you are doing something which most people in the world are not aware of. Finally you're going to have a small problem which is going to still, poke at you all through the day. And that is important, because finally you will get a solution, and then that happiness that you get, that is incomparable. Just incomparable. And in fact, you nowadays, when you go abroad, there are, especially in Europe, you can get jobs. They want people. Canada, you can actually become a Canadian citizen nowadays if you are a skilled professional. There are opportunities. So whatever it is, I'm trying to tell you, get out of that comfort zone, get out of that flow of being an engineer and a doctor and all. We need that. Doctors too, we certainly need. We have too few doctors, but we have lots of engineers. This is the most important thing. This is the most important thing. And this, if you know, then I can tell you will be successful in whatever you do in life. So there was, I think, some discussion, this previous talk, so beautiful genetics, fantastic things, right? So I think there was some little question that had come about white flowers which are different colors in the same plant. Suppose now you see a small plant where there are red flowers. There is only one white flower. I am making this up. I am not a biologist, not a botanist, though, chori do. Suppose you see a white flower. You want to question why is there a white flower in all these red flowers? What is it? How do you know? What is the reason? And that getting interested is the first step of being a scientist. That itne lal hai, why is this white flower? Why is it there? What will you do? What will you do? My challenge is I will find out why is this white flower there. First thing to do would be to observe. Are all the flowers just very red and this one flower is only white, or is there a gradient? Do you have some with a red, some with a reddish white, some with a whitish red, or do you find them all white and red? All red and just that white. That is an important critical measurement. Second step, you observe, measure, analyze. What will you do? Probably do a genetic assay, right? Probably there's some albinism, there's some albino gene which has got manifested. You find that out. You find that out and then you infer. Oh, so this is the reason. This is the reason that there is one gene which has got triggered, which has expressed itself, and therefore this is the reason. And then you predict what can, so this is the probability of this gene getting expressed. So if I have now this many generations, what is the probability of having other white flowers? Can I actually do a mixture? Can I do a crossbreed or something and try to express that gene, force that gene? Can I have now a tree which has all, whatever plant which has all reddish white flowers? But the critical part is these 5 things. And believe me, whether you are a scientist, whether you are a banker, whether you are a singer, whether you are a football player, everything in your life, these 5 things will come in when you want to understand, when you want to solve problems. Suppose your electric bill is very high at home. You actually figure out finally that the reason is your taps are bad. There is continuous water leakage in the house. You get a plumber, all the taps are fixed, so your electricity bill comes down. That only will happen if you do all these. That has nothing to do with science, but your father will be very happy with you. A genius ladki. Right? Anything you do in life, these 5 are crucial. Standard path, I'm not going to bore you with that, but this is critical. Critical. If you want to do a PhD, abroad, not correct. This is something that I did. Bhatnagar, have you heard of the Bhatnagar Award, SS Bhatnagar Award, that's like the holy grail of physicists of scientists in India. Very, very big thing. It's like, I mean, it's like a far short, but in India it is like a Desi Nobel Prize or something. But of course, I'm not even comparing those. From 2012 to 22, 10, 11 years, 21 Bhatnagar awardees, 12 experimentalists, 11 with PhDs from India. And why is that? The reason is that there are many, many things in India when you do experiments which are not in your control. You will have your account section lose your purchase order. You will have things go defunct. Nowadays, buying things is so much difficult. If you do a rosy PhD at Waterloo and at New York, and then you call up a supplier and they give you your instruments, you come here and suddenly you realize that aisa nahi hota hai. Everything is so different. You call a supplier, he'll tell, sir, finally the whole process finishes in 1 year. Right? So that I have seen. In ISC, we have seen people coming from abroad 3 years after they have left because they just could not find enough. They were just suffocated. They thought that they were not used to this country. So finally, what do we say? Liberty, creativity, fraternity, be hai, but contribution and satisfaction, that is critical. As I said, I don't need to re-emphasize. As I said, 10, 14 years I've made a lab and I've learned 3 new things this semester itself, up to now. I don't know what is left. Much, much better. This is where I will go on to the last part. This. I have no qualms in saying that in my research journey, I have been very lucky with accidents. Serendipity is the word. Have you heard of the word serendipity? Happy accident. At least 2 things we have done is something that I would not have done had I been. So optical tweezers is not something I've ever done in my life. My PhD was on a very different thing. Very, very different thing. My postdoc job in GE was very different. I was making sensors, optics-based sensors. Optical tweezers, the first time I saw, I mean not saw, okay, our lab had got that, but the first time I actually worked with them, I actually saw a particle trapped was in my lab. And that too, the student trapped the particle when I was having lunch. I came up and I wanted to hit him actually. This is something that I'm going to tell you. And so there are these 2 things, right? Both of them together have produced some now 5 PhDs, many masters kids, masters graduates. So what we were doing, as I said, we have this microscope which has an objective lens which focuses light into a small sample chamber, which is, you have seen a microscope slide, right? And you've seen cover slips. So cover slips are about 25 millimeter by 15 millimeter, something like a rectangle. And a microscope slide is like what? This type, 3 inches by 1 inch type. So what we do is we take this cover slip, we put some 40-50 microliters of sample, and then we just put the slide on top, take the sandwich, it sticks because of the surface tension of water, and we put that on the microscope stage. So what we were trying to do was something very high-fi. So ISER, the motto of ISER is interdisciplinary science. Big words, but actually it works. It works because we don't have separate buildings for physics, chemistry, biology, and maths and earth sciences. They are the 5 things we have. We have humanities and other things also, but very small. All of that is in one building. And the days I'm talking about, we were living in a bhara building that was of the Haringhata farm there. So we used to have tea. There was a shanty tea shop, Ganeshda Chai Dukan. We were having tea with a friend of mine in chemistry who had just joined 6 months earlier and was having no money. He was not sure what to do. So then we were thinking of, okay, we'll do something very interesting. So he works on these things called soft oxometalates. So these are some polymers which when you put in water, they become like this jelly-like substance, but then they're semi-solid, right? And we were thinking of doing something on shape memory. Shape memory means you have something like that, you put some stress and you leave the stress and it will come back. Okay, very hi-fi stuff. So what my student was trying was to get these soft polymers. So if you put these soft polymers which come as powders, if you put them into water, they self-assemble and form these aggregates which are all squishy and jelly-like. So we are doing that, very, very old. This looks like archaic footage, like this is 14-year-old footage, 13-year-old. So see, we were trying to focus light onto that, and when we were focusing light, what was happening was these were breaking. I don't know whether you can see, but you will also observe that this is the focal spot. You see this bubble coming up, this bubble. My student was completely irritated. Arey bhai, whatever I try to do, this bubble comes. Arey bubble ko hatao. What do we do with this bubble? And then he came and said, sir, I am going to leave my PhD. I am trying to do this. Whenever I focus light, there is this big bubble that comes. And then he told me something more interesting. Sir, there is something very odd. Ki when I am trying to remove the bubble, what do I do when I say remove the bubble? I move the laser spot. That is, I just translate the microscope stage. This bubble is following. Very dangerous bubble, right? You're trying to move it away, the bubble is running with you. Exactly like this. This is a stage. I am just translating and this bubble is running. Bubble is running. But see behind, what is this? You see this thing, these two? This is like a, do you realize this thing? See, the bubble is running. I am translating, the bubble is running. You see what is behind? It's leaving like, it's like the wake of a jet plane, right? You've seen jet planes going, right? They leave those two tracks because of this, because they are traveling at supersonic speeds, right? The nucleation, they cause just nucleation of water vapor, just the water because of the sonic boom and all that. So you see this exactly like that? This was a new field that we discovered because of this. You know what happens? This is a new term we have called this. What we are actually doing is we are making patterns, permanent patterns of whatever we had put into that dispersion. So suppose you have taken some powder of some object inside water and you are basically taking this on some slide, you are focusing with the laser. As I said, laser is very intense. So when you have that absorbing, so what happens is if you have, so very critically, the wavelength of light at which I am using, that is the wavelength where this particle is absorbing. These particles which are dispersed in the water, they absorb that wavelength. Now if I have a dispersion where all these little absorbing species are, floating around, some of them will go and stick on the surface, right, on the glass. They will stick. Now if you focus your beam on that sticking thing, what will happen? That thing will get very hot because it's very small spot, and you know how small that spot is? 1 micron. What is 1 micron? Your hair, you know how thick it is? 1 strand of hair. Any idea, somebody? No one knows thickness of hair. Average 100 microns. So I'm talking about 1/100th of a hair. That is how small my focal spot is. And now if you divide power by area, so if you have like 10 milliwatts, divide by that pi r square, big spot radius, you will get gigawatts per meter square. Extreme, it will burn through your skin, come out. That hot. So what happens is, because this absorbs, the water gets very hot locally. And it cannot stay as water, it boils. But remember, the rest of the water is very cold. So what happens? You get a bubble. You get a bubble, right? Now, this part of the bubble is hot, that part is cold, right? Because the laser is here. Now, if this is hot and that is cold, when it's hot, the bubble wants to expand. When it's cold, the bubble wants to contract. When it expands, it drives away the water around. When it contracts, it pulls in water. So there is a flow. And that flow drags in whatever species you have dispersed in that solution, or dispersion, suspension, and they come and accumulate at the bubble corner. So this screen is resolution is bad. So what happens essentially is that you get material that is deposited at the corners, and when you now pull the laser or you pull the translation stage, the spot moves. Now when the spot moves, the bubble cannot be there because the heat source is gone. So you can either have a new bubble or that bubble can move. Now new bubble is energetically more costly. So therefore this bubble moves. And now you move it in whatever way you want. And the moment the bubble moves, this material comes in at the corners and it keeps on coming. So I can write ISER. Not only can I, so what does, this was the paper that came out in 2013. We were the first in the world to show this. Of course, another thing is that people don't acknowledge that, especially abroad. That is a different story. But at the same time, nobody can, we know that we did it. So these are all the things that what the world has done. This is ours. Can you see this? Can you recognize this person? Resolution is bad, but some vague idea? Yes. This has been drawn using that. So what actually controls, so what you have is, if you move the bubble in a straight line, you will get a pattern, a linear pattern. The thickness depends on how big the bubble is. And how can you make the bubble bigger? You use higher laser power, you use high concentration of material so that for the same time during which the laser is on, the bubble gets bigger, and you move the bubble very slowly and allow it to become very big. Right? But the applications are fantastic. You can, so we actually have said in a review paper that you can pattern everything mesoscopic. When I say everything, I am not joking. It is indeed everything. People have made circuits. This is our work. So we take conducting polymers, plastic electronics. So these are, so I can actually draw circuits. Circuits mean I deposit something which has high conductivity. And what we have seen is in the process of depositing, I have actually increased the conductivity of the species. It's quite phenomenal, right? So people have done all silver. This is polyaniline, there is silver. This is very nice. This was actually done using a smartphone. So you know what they did? The translation stage was moved by an app drawn in the smartphone. So whatever you design, you draw with your finger, that translation stage will move like that. Okay, this is all engineering. It's not a big deal, but nice, right? All kinds of things. This showed that if you move very slowly, these were actually nano dots, quantum dots. So if you draw them slowly, so if you move slowly, then more and more dots accumulate for the same time, and then the fluorescence properties change. This is something interesting, but you can do all kinds of different colors, right? Catalysis. So you actually print catalysts, and you can actually run catalytic reactions parallelly. This is the most interesting thing. Can I actually print living matter? Can I print viruses? Can I print bacteria? That is a bit of a problem because these bubbles are very hot, right? You will think that any living matter, any bacteria will fry. Fried bacteria ho jayega, fried virus ho jayega. So is there a way out? Yes, there can be a way out. You use small bubbles and you go fast so that the bacteria will not even realize it's so hot. It will be pulled in, but by the time it is there, the bubble is gone. Right? And we actually found our biologists friends who did it with us, they actually did not know. They did not know that if you keep this virus, we actually have patterned the virus, if you keep it immersed in water for 10 seconds at 100 degrees, it does not die. He had no clue. I did not know. Because why will you do? Why will you take a bacteria or virus and keep it at 100 degrees? But they did it and yeah, it does not die. And we are doing it in milliseconds. We are doing it very fast. So look at this. This is bacteria, E. coli. So what we do is we don't pattern E. coli directly. We actually pattern something first to which the E. coli sticks. to which the E. coli sticks. That we pattern, and then we run the bubble on this part, and you see E. coli sticks nicely. This is on H1N1, swine flu virus. You can see nicely patterned, and what we have is these viruses have some fluorophore. So when you illuminate with green light, you see fluorescence, blood group sensing. So you can actually, this was our student's blood. So with research, for research you need blood, sweat, tears, everything. Okay, so you can see that this was whatever blood group. You see this line here, so this is actually some kind of coagulation has happened. So this is the blood group. This is the final thought of the future. Okay, that you will have this microfluidic assay with different pathogens being patterned and parallelized sensing for all of these. So think of this, if it is possible sometime in the future, you can actually make one detection chip, just maybe put a bit of saliva, and it will tell you whether you have COVID or malaria or whatever in one test. So this technology completely has happened in my lab our lab at IACS Kolkata. And because we just pursued things which we did not understand. Okay? This was the paper where if you want to read about it, it's there. A little bit of IACS Kolkata, I am not going, this is I'm done, right? So IACS Kolkata, this is looking very glamorous, right? But this is actually a swimming pool. Indeed a swimming pool. So IACS has 5 or 4 programs. Very few people have heard of IACS Kolkata, which is still bad, but our flagship program is the BSMS thing that we have after our school exam, class 12. You can get in either through IIT or JEE mains, or by just, there's an exam for top 10% or top 30% of the board exam that you give. There's a written exam then. And we have PhD, PhD, everything. Interesting part is that we are doing well. We are getting faculty, our PhD students placed in good places. We have had, it's a good place. We are doing well. If you want to know more about it, visit our webpage. Okay? This is also my final slide, and this is something, these we call, you know whom these are, but if I tell you that these are Bengal's first innovators, will you agree with me? Will you say that these are Bengal's first entrepreneurs? That too you'll say no. But why not? This guy, we think of him as spiritual, but he actually obtained funding in those days. And he funding for his trip to the US. And he started the first NGO in India, which is doing fantastically well now, social NGO. Social NGO. This guy, we know his songs, we sing his songs and poetry and upanyas and all that. But do we realize that the education system that he had made in Shantiniketan was unparalleled, unthinkable during those times. Now people are copying it in Finland and Japan and Germany and all that, whereas Shantiniketan itself has deserted it. Where there is no class. A person can be in class 9 in history and class 5 in math. That is exactly the model that people do in the West. The first education and entrepreneur in the country. Of course, this guy, it's fine. Bose and Prafulla Roy, of course, perfect. But RISE Foundation is something which we have grown with the help of the government of India. We have 21 incubators. You know what is it to make a startup? You have an idea, you think it's good, you have a dream, you want funding, we can probably give you funding. Because finally, science and business can be success with happiness. Thank you. So I guess people are hungry and I've probably taken a long time, so I don't think too many, whatever, yeah. I'm free for questions.
Any questions for sir, please? Kimba. So, sir,
Yeah.
Thank you very much for sparing your valuable time with us
Pleasure.
today.
Okay.
Okay, we shall now be, sir, please request you to take your seat in the audience.
Yeah.
We shall now be dispersing for lunch, but before that, I have a few announcements to make. Firstly, Vivekananda College, Thakurpukur, you're requested to collect your lunch coupon from Ms. Deepanita Chakraborty in the registration desk, and also Mr. Robins Kumar Shah, are you there? From Mumkin Shraddha College, you also have not collected your lunch coupon. Okay, so we will be having the lunch in the open air theatre, and our organizing committee members are outside to guide you there. And after the lunch session, we are also having our photography and videography team. You can have a photo session in our campus. Okay, they'll be also guiding you. And after lunch, we'll be gathering here sharp at 2:30 p.m. Okay, after that, we'll be having a demonstration and lab visit. So all of you, please assemble here sharp at 2:30 p.m. Okay. So welcome back, everyone. Now we would be having a third invited talk of the day, for which we have Professor Arindam Ghosh from IISc among us. Sir, may I kindly request you to please come on the dais, and Professor Debashri Ghosh, ma'am, I would request you also to kindly come up. Anushmita and Payal would request you to please felicitate sir with a bouquet and a replica of the Raman spectrometer. Ma'am, would you kindly introduce sir to us?
Welcome back, everybody. It is my pleasure to introduce Professor Arindam Ghosh, who is an experimental condensed matter physicist in the Department of Physics at IACS Bangalore. He actually obtained his PhD from IACS in 1999 and did his postdoctoral research at University of Cambridge, after which he joined his alma mater, IACS. He is one of the leading experimental physicists in the area of atomically thin two-dimensional semiconductors for building new generation of functional electronic, thermoelectric, and optoelectronic devices. And I mean, I can go on a lot about his research, but maybe it is better to hear from him about his research. Just to give a little bit of more info, he has many accolades, among which he has been awarded the Bhatnagar Prize, Infosys Prize, and many more. Again, I will not bore you with all of them. It is an absolute pleasure for the research community at IACS to welcome Professor Ghosh as the speaker of the National Science Day 2024. Professor Ghosh.
Okay. You can hear me now, right? Okay, good. First of all, thank you very much for the kind introduction, and also Shubhadeep for giving me the opportunity to come here on this important day. There's a lot of events going on, and I think it's good that we connect at least on one of these days to what's happening in science and technology in India in various fields. And I think today it's extremely important that we engage, because the awareness about science, that we should increase the number of people who are doing science in India, is essential to better the quality of our lives, is becoming more and more important. So I think it's good that we are all here. And what I'm going to do today is I'm going to tell you how science and technology have moved together. That's the most important thing, right? Today, science is important, understanding fundamental processes are important, but how to take the fundamental understanding to actual implementation of technology, and what are the key aspects of science that we must focus on, is essential to understand. Okay? So I think what we will do, we'll try to do a little bit of history, and then I'll go to a little bit more detail. I don't have to finish everything that I have got here, but let's have it interactive. Please do, stop me if you have questions, and no question is bad question. So please ask me questions whenever you want to. So if you look at the way science has developed over the last 100 or 100 and few years, few decades more, is if you look at the revolution, essentially the industrial revolution that started in the late 1880s or early 1900s, it's about heavy machineries, right? It's about the new manufacturing units that started, and that really started changing the scientific ecosystem and the technology ecosystem in the world. Once that started, we started having very fancy things like the cars, for example, the machineries like heavy engines, the powerful engines of Rolls-Royce to helium liquefier, and that fed into the scientific ecosystem as well, the new scientific discoveries. The moment the compressor of the helium liquefier was created, it didn't take very long to liquefy helium, and once we liquefied helium, we discovered superconductivity. And this is, by the way, I do not know whether you would know between these two people who is the Kamalin Onus, who discovered, who's in charge of the lab, in Leiden, who discovered the superconductivity, the liquid mercury, electrical resistance of mercury going down. Okay, so this is, by the way, is Kamalin Onus. The story goes that when actually the resistance of mercury fell, he didn't believe it, and the person who was working was fired because he thought that it's a bad experiment, but it turned out that's not the case. Right. Okay, so not too long after that, we started understanding how to make fancy and complex machines, which will look deeper into material science, in which we started looking at atoms, which started looking at molecules, and for that we needed new kinds of microscopes. So instrumentation, the technologies of instrumentation led to science, and that science again led to instrumentation. This is the first transmission electron microscope in 1931. And then not too long after that, von Ardone showed the scanning electron microscope in 1937, with which we first started looking at tiny, tiny structures. Today's nanotechnology, what you see today in various materials and various systems which we make silicon MOSFETs, are all in nanoscale, a few nanometer in size. They wouldn't have been possible unless and until we could see the structures that we are making today, right? And these are 1930s to 1940s machines that allowed us to do this. But in addition to, when we are able to see the system, but we have to make those systems, right? And those systems started coming with systems like high vacuum system, in which you could measure, or you could deposit thin films, okay? And these thin films, they are not at all unimportant, although if you look at the first early vacuum chambers in which you deposit thin film, you take a metal, you heat it up with the filament, and the molecules evaporate and they get deposited on a substrate. Okay, you might think that, okay, this must be fairly straightforward thing to do. What's interesting can happen? If you look at the 2007 Nobel Prize by Peter Gruner and Grunberg and Albert Firth, they essentially deposited thin films. They deposited thin films of two magnetic materials and in between an insulator, correct? And those, that tunnel junction or two magnetic systems which are separated by a non-magnetic thin film is essentially the science which they were looking for, how to look at the electrons which is going from one magnetic layer to the other through the non-magnetic layer, and whether by relative orientation of magnetization that you can control the current. Now, this was a pure scientific quest, and that scientific quest led to technology. Today's hard disk drive, for example, the sensors and the magnetic random access memories, that's going to go probably in a big way as a low dissipative memory structure, both for reading as well as computing, are all now in an entire field of spintronics in 2024 is dependent on this kind of vacuum units on which you could do thin film deposition, okay? So what's important to understand is that designing the instruments, designing the materials, and giving them unique structure is the first step towards getting extremely important for next generation technologies. This is what I wanted to impress upon with this short introduction. Of course, if you look at in addition to vacuum chamber that I showed you in the previous slide, this is in 1960s. Today, vacuum chambers are relatively more complicated. At that point of time, you could reach vacuums of about 10 power of minus 6. Today, we can go to vacuums of 10 power of minus 11 millibar in a vacuum chamber, which is essential because now you can make extremely high quality depositions, and for silicon and semiconductor industry, that's the first step to do. Because if you take a look at the semiconductor electronics since 1947, this is John Bardine's first field effect transistor on germanium. Look at the size of it, right? That was a bulk, massive one. And today, in an Intel Core i9 processor, you have 1.2 trillion of this type of field effect transistors buried inside. Now, this wouldn't have been possible if we had not got fantastic deposition, fantastic material synthesis, and characterization technologies. Okay, right. But that's very good. These are all that over the last 4 or 5 decades of experimental and technological advances. What about new technologies? And that's what we are here to talk about. The new technologies, we more or less are relatively, we more or less have a direction. What are the grand challenges to humanity in terms of various technological fronts? We need energy solutions, particularly hydrogen and batteries, storage, as well as generation in the green. We need genomics, the personalized medicines. These are key challenges. Then we have got artificial intelligence, machine learning, big data. I was just recently told that ever since the ChatGPT and these artificial intelligence the protocols came up over the last 2 years to 3 years, the computing requirement has increased 175 times. Everybody, this AI computation is far more taxing and on our computing resources than what Moore's law earlier, which essentially said that our number of transistors in a chip increases by a factor of 2 over about a year or two. That essentially is simply not enough. No matter how well the technology moves, the computing requirement increases much, much faster than what Moore's law of technological advance can do. So clearly, we require disruptive solutions. We need new solutions that will help us keep up with all the artificial intelligence and machine learning algorithms implementation. And this is where emergence and the new technologies would come to play, and I shall give you a couple of examples. Okay, so what I'm going to do is that I'm going to give you a couple of examples from what is needed for what's coming up as quantum technologies. So what is quantum technology? I'm sure many of you are from physics background, or even if you're not, it doesn't matter, because quantum technologies is one of the areas which sort of transcends from one area of science to another. There is a highly interdisciplinary area. So quantum technology is essentially an evolution of our understanding of quantum physics over the last 100 years. Okay? This is Erwin Schrödinger in 1900s, and then somewhere around 1980s, where maybe I can go that side. It's easier for me to see what I'm talking about. Okay. We have Richard Feynman saying that, okay, there's a lot of rooms down there so that you can utilize them. The whole concept of computing using quantum mechanics that started coming in. So essentially, the understanding of matter with quantum mechanics over the first 70 to 80 years, and then engineering of quantum physics into new devices led to this revolution of new type of devices. Right? So this is something which is the future, in which we would like to see some of the hidden aspects of quantum physics being used as an engineering strategy or engineering platform to get devices that are disruptive. It means that it's completely different. A standard classical physics simply cannot, or classical devices simply cannot perform those tasks. They can be enormously sensitive. I'll try and give an example of that. And they are really, really fast in at least some applications. Okay? Now, these three is usually dependent on some of the key aspects of, three of the key aspects of quantum physics, which was never used. You see, we have been utilizing or using quantum physics for understanding materials, understanding our physical properties of systems, using the Schrödinger's equations and various other methods where it's the electronic wave function and the phase are of important quantity. Whereas now the new quantum era is using this new properties which are relatively non-intuitive properties like entanglement, superpositions, and topology. I'm not going to say much about this, but I'll try and give you some example of the first one, okay, and see where it can help us. So this is a rather non-intuitive property. There are a lot of very interesting articles on what quantum entanglement is, and it's not something that we can even, there are problems of understanding the reality, and that's the fundamental questions that entanglement imposes. So we'll not go into that detail because it has been told a long time back. If you think you understand quantum physics, then you probably do not. Okay? So that's why we will not even try, and this is what David Mermin from Cornell University said a long time back, that if you don't understand, you just shut up and calculate, okay? And by the way, there is a very interesting statement, because many people who do quantum physics calculations, they do the opposite, and hence there was a statement, and okay, if you're doing quantum physics calculations, you calculate and then shut up. But anyway, I think this is one of the important things about quantum physics, that we have understood quite a bit, so it's time for us to do some engineering with it, okay? How the evolution went, it's not too far from the way our technological evolution went over many years. 1900s, black body radiation, I'm sure many of you have already told, and then, or read, then photoelectric effect in 1905, which sort of told us that the quantum aspect of light and energy. Then we have the wave-particle duality in 1923, Schrodinger's equation in 1925. Then came EPR paradox and the thought experiment in 1935. This is roughly where people were understanding that quantum physics is not going to be particularly, it's not straightforward. There are challenges to have an intuitive understanding. 1957, BCS theory essentially showed that quantum physics can explain some of the new phenomena at that point of time of superconductivity, and hence people started believing that yes, quantum physics probably is correct. Okay, we have Bell's theorem in 1964, essentially showing that okay, can you differentiate between quantum physical concepts, entanglement, etc., from the other possibilities of hidden variable theory and various other competing arguments which tried to understand the physical reality. Then lasers in 1985, and finally we have now supremacy of a quantum computing in 2019, essentially paving the way of engineering quantum physics and the hidden aspects of quantum physics for real life applications. Okay? So you see, it's essentially a journey of more than 120 years. What are we supposed to get if this promise of engineering quantum physics actually becomes fruitful? Well, I mean, we have fantastic possibilities of making quantum clocks. For example, if you can make clocks with 10 power of minus 19 stability, that means 1 part in 10 to the power of 19, which seconds, which means that probably our synchronization between different GPS systems can happen without going through the satellites. So navigation, you can Ubers or you go through different roads, which will not require a satellite, is one possibility. Of course, that will require the clocks to be sitting in our cars, which is not very easy. We have simulators, we can have drug design, we have sensors, and this is something which I'll give you an example of, right? So we can have a new generation of sensors, which in principle can go for exploration of natural resources. It can also have a precision measurement of plate tectonics. For example, can you predict earthquakes? But today, there is no good way of measuring the tiny movements of continental plates because it's so small. But can we have sensors which can actually lead to measurement of slight variations in the gravitational constant at the 1 part per billion accuracy, so which can tell us that there is something which is happening and whether that could be modeled. So these are natural promise and challenges that quantum sensing gives us. We have medical imaging. For example, if you can measure tiny movements of the plates, you might want to also measure tiny electromagnetic waves that is emitted by the brain, for example. A small magnetic field or small electric field that the charge motion in our brain can give. So we have medical imaging and new way of diagnosis. That is something which is possible. Of course, secured quantum internet, and of course quantum computing, in which you will want to look at a new way of optimization, new ways of predictions of financial markets, and so on and so forth. These are all way down into the future. But there are certain things that are certainly possible within next few years or so. And for that, we require certain very important tools And this is where the new technology platforms for this new technology must be built on. I'll give you an example of quantum entanglement. I told you I'll do that. Let me try to explain this to you with a little bit of oversimplified example, but it's not too bad. Suppose you have an apple, okay, and you want to measure the size of the apple for whatever reason. I think it's very difficult to do this if you go to the market, but still, let's suppose you want to do this. And what you do to measure the apple, you want to use a scale, right? Say a scale like this. If you want to measure the apple very, very accurately, then you try to measure it many, many times. And many, many times you use many scales, not just once. You 1, 2, 3, and you keep on measuring it. And suppose you measure it n times, correct? If you measure it n times, then what you get is a standard quantum limit. It means essentially the error in my measurement will be given by each measurement minus the mean whole square divided by the number of times it has been measured, right? So this is the definition of standard deviation. Now, which means that my error will go as 1 over root n, where n is the number of times I measure it. Turns out that now you do something really funny. You take all the scales and you make them quantum mechanically entangled. There's some hidden property. The quantum mechanics comes into play and says that all my scales are quantum mechanically entangled. It turns out that in that case, the error is what is called the Heisenberg limit, doesn't go as 1 over root n, but it goes as 1 over n. Okay? Which means now, if I do the measurement million times, okay, suppose I measure it instead of 1 or 2, I measure it 10 to the power of 6 times. In which case, the error in a quantum measurement is at least 1 over 1,000 the error in the classical measurement. That's the key aspect of doing a quantum technology as opposed to a classical technology in which you are able to get very high quality measurement result. So what kind of scale are we talking about here? One of the example is, say, each measurement is done by a photon, 1 photon. You can ask the question, oh, okay, to tell me an example, how can a measurement be done with a photon? All right. Suppose you want a radar, okay? A radar is what? A radar is, you have a system at the ground, and there's an aeroplane which is flying in the sky, and you essentially fire a photon which reflects off the aircraft and comes back, right? Then that's a radar. And once you detect that photon, you know there is a aircraft or something moving in the sky, and you detect it, and that's where the military or aviation industry, that's how it looks at. Now, if there's a lot of noise, for example, cloud, and the number of photons that go in are very different from the number of photons that come back, then it's very difficult to detect the aircraft. Now, tomorrow, suppose I do a little bit of engineering, and instead of making a classical radar like this, I make a quantum radar. A quantum radar essentially is, instead of 1 photon which is going and firing the aircraft and coming back, I use 2 photons. But these photons are quantum mechanically entangled photons. I told you scales, so you have to understand that the scale is a very different concept in quantum physics as opposed to a classical scales that we see. Scale is a measurement, right? I mean, that's a measurement platform or a measurement strategy. So now I've got a pair of entangled photons, and one of the photons I keep to myself, and other photon I fire to the aircraft. When that photon goes and comes back from the aircraft, I measure them simultaneously and compare whether these 2 photons are correlated with each other. If they are, then I know that something has been reflecting them. And because they are quantum mechanically entangled, they are non-local, and these lead to a significant reduction in the noise in detecting the aircraft which is flying up there. And there are numbers how much noise can be reduced or something of that sort. Well, but the point is that this is a unique way of going below what you call a short noise limit, or the basic fundamental limit of measurement that classical measurements will be bounded by, okay? Very good. So question is that what is a single photon then? Okay, let's go in further, deeper and deeper about what this technology requires. I gave you an example in which 1 photon or detecting 1 photon is essential because you are talking about 2 photons, right? 1 in two entangled photons, which means I need to be able to measure them. I need to send it across to the aircraft, get it back, and then I have to measure them. Now, how easy it is to measure a single photon? All right, it's not very easy at all. It's easy to talk, easy to say. It's not easy at all. For example, a single photon means you need to have an energy resolution of your detector to be about 10 power of minus 19 joules, right? H cross omega, H is 10 power minus 34, W is 10 to the 15. So typical energy of a single photon is 10 power of minus 19 joule. How much of an energy? Do you have an idea here? Suppose I have a candle, okay, in this room. Switch everything off, a candle. And this candle emits a certain amount of energy. And that energy is called a candela. A candle amount of energy emits. A candela is typically about 18.3 milliwatt per 4 pi r square. So around a circle, it emits about 18.3 milliwatt. Very good. So now suppose you make this candle, light this candle up on Earth, all other light sources switched off. So then you have a moon about 384,000 kilometers from Earth. It turns out that the number of photons is 1 in an area of 1 square meter, about 1/3 the distance to the moon, which means if I have one candle on Earth, there would be 1 photon per unit, per unit time, that is per second, will go through 1 square meter, 120,000 kilometers from the Earth. So that is how small an energy is, 1 photon's energy. And we need to measure them. We need to measure it experimentally to do a quantum physics experiment, right? So that's certainly one of the challenging tasks, but it's not enough just to be able to measure 1 photon. There are a lot of other challenges in making them useful for quantum physics or quantum technologies. For example, you need to measure such photons which at a very high rate, because it's not just 1 photon per second. You need to measure 10 to the power of 6 photons per second or something of that sort to make it a useful technology. Then you have to have high efficiency. You need to measure them 90% of the times, because sometimes what happens, the photons go in and you simply don't record it. That's not allowed. You need to measure this with extremely high efficiency. You have to have very large spectral range, and this is a very important ability, number resolving ability. For example, if I have a laser, or if I have a light emitting diode, and I give it a pulse, every pulse will have a certain number of photons. It's not necessarily 1. And the number of pulse is usually a Poissonian distribution, depending upon the intensity. So you need to know how many photons are there in a pulse in order to make it useful for quantum information processing. There are quantum computing protocols which depend on photonic processing, and they require this number resolving possibility. And it's extremely difficult to design detectors of this type of capabilities. So today the technology is, there are several technologies that are being proposed and followed. They all have some kind of a limitation. For example, photomultiplier tube, in which you put light in and that interacts with some vacuum valve or something of that sort. Now these are very fast and etc., but their efficiency is only 2%. So out of 100 photons that come in, only 2 are getting detected. It's not good enough. Semiconductors have got avalanche diodes, for example. They are one of the important technologies. And problem with them is that they are very good. Their efficiency is 30%, but the number resolving capability is not there, which means that you cannot, they are called click detectors. That means if this is 1 or few photons, it will say yes, and there is no photon, it will say no. But it cannot say, oh, okay, this pulse of light has got 5 photons, this pulse of light has got 3 photons. It cannot say that. And that's the problem. You cannot use them in photonic processing. Yeah.
We are looking for the reflected photons,
Right.
And here you are talking about entangled photons.
Right.
But in that case, detection is not only the photon, but the entanglement also.
Correct. No, so in that you do an optic interferometry measurement. The detection of entanglement requires 2 detectors of that type and measure the coincidence measurements, right? You see whether the two photons come simultaneously or they do not, because the coincidence measurement will tell you if the photons are entangled. What I'm trying to say here is that not the entanglement, but the very fact that how many is, in a photonic processing protocol like Gaussian sampling and various other methods, in which you need the number, information, how many photons are there in a single pulse of light. It has entanglement property will come afterwards.
Photon count.
It's a photon counting, right? Just photon counting, okay? So today's technology is primarily this kind of photon counting technology is based on superconductors. It turns out that if you take this nanowire superconductors here, when the photon falls on the superconductor, it locally heats the superconductor, and the heat converts the superconductor into a normal metal, and the resistance changes. Now that seems to be a good way, okay? And this good way gives us the probability or the number information that it is number resolving, but the problem now is that it is usually very, very slow process. So we need these detection to happen at megahertz, at gigahertz, 10 to the power of 6, 10 to the 9 photons per second. Most of these detectors are not possible. They can't do this, okay? So there is requirement of new strategies of functionality designing, okay? And this is where the emergence comes into play. Can we use this, a whole new strategy to make functionality or design functionality from bottom up? So what is the idea here? Can we engineer emergent functionality with materials? The idea here is that we would like to combine materials with different physical properties to achieve a new and disruptive property or functionality that does not exist in individual components. Okay, I'm in Kolkata. So what exactly it means is something like this. I don't know. Anyone recognizes what is this? Somebody? Right, right. Or this. That's not Hasjal. What's this? Okay, right. So what I'm going to talk about, the basic idea is this. How do we design? Of course, at that point of time when Sukumar Rai actually wrote Abul-Tabul, I don't think he was thinking about using them as single photon detectors, but we can take this strategy. And when this material design strategy actually came out, actually it's not too different from what we are doing today, okay? I'm going to give you an example with this, and I'm now going to go to a little bit more. How much time do I have, by the way? 10 more minutes? Okay, so I'll give you one example, and essentially giving you the idea of the whole concept of designing materials bottom up with overall concept like of this sort, that we take two chemically or physically different type of system, combine them to get a functionality which didn't exist in either of them, okay? Take graphite. So this one material is graphite, okay? We know what graphite is. It essentially is a stack of large number of layers. And one of these layers is called graphene. Have you heard of graphene? How many people have heard of graphene? Raise your hands, maybe. Okay, good, excellent. You've got plenty, that will do. Okay, sorry? Questions? Okay, maybe later. So you have got graphene, correct? Many people work on graphene. One of the unique aspects of graphene is that, or this class of materials, is that you have a very, very weak coupling between the layers, and this is why they are called two-dimensional materials. The coupling between the two layers is about 1,000th or 100th of the standard covalent or ionic bonds, which are made from, say, three-dimensional solids, something of that sort. Because of which, now it's possible to make devices with just one layer. And this is what I do, and many people in the world do as well, where you take a layer which we press on silicon oxide or some substrate, make electrical contacts, and that strategy essentially of how to make this electronic devices with this was discovered in 2004. That led to a Nobel Prize for these two people from Manchester University to show that it's a very, very easy way of making extremely high quality or completely flat, physically, the fundamental limit of thickness of an electronic channel, right? It led to, and this is one of the key aspects in carbon has been quite an interesting system, and not only graphene, but fullerenes got Nobel Prize from Kroto and Smalley and Kerr in 1996, and of course Ijima's carbon nanotube got Kavli in 2008, and of course, and graphene, of course, in 2004 was discovered, and Nobel Prize in 2010. This is a very good thing, but actually the graphene started much, much earlier. Way back in 1967, there was Philip Wallace in Canada, a Canadian scientist, who sort of said graphene is a very interesting system in which you take a look at its electronic properties, where it's a pi bond, pi electrons, and there are 3 sigma bonds in the plane, and pi electron is the reason why it conducts electricity, and this led to a very interesting electronic energy versus momentum relation, which is like photons, which is E is proportional to K, but the speed of electrons is of course much smaller than that of light. The difference is that if you take a Schrodinger's particle, which is a massive particle, E is proportional to square of momentum, whereas in graphene it is proportional to K. And that led to a very interesting set of properties. For example, the electrical mobility of graphene is much larger. It can easily conduct electricity much, much better, at least 3 to 4 orders of magnitude better than a standard silicon MOSFET, even though they are both two-dimensional systems. Okay? So electrical mobility of graphene is very, very much better. And that is why you can make extremely fast electronic transistors with graphene. Some of the highest frequency transistors could be obtained at small gate length using graphene as a platform. In addition to that, because the sigma bonds is extremely hard, it's one of the most hardest system in nature. For example, for a similar thickness, graphene is more than 200 times stronger than even steel, right? Because of the strong sigma bond in the plane. So there are these very good properties, and many people are using graphene for various reasons. But it turns out that graphene is not alone. Chemically, there are its cousins or layered systems, which can be a beautiful semiconductor, like the molybdenum disulfide-based materials. They are semiconductors. There are insulators like the boron nitride, etc. And there are superconductors like high TC superconductors. They all fall under this layered structure or layered family. So the question that we wanted to ask is, can we do something which is mixing and matching? The semiconductor-based molybdenum disulfide was discovered or made your transistor from EPFL way back in 2011. And also exactly the same kind of electronic devices could be made with molybdenum disulfide. There are more than 1,800 such systems. So really, if you want to start working on it, you will need about 3-4 lifetimes to go through all the systems of two-dimensional origin. But anyway, so the question is that we have several of these systems which are 2D, but they are chemically different. What can we do if you couple them? Okay, so this is an old saying, but anyway, the idea is that if we couple them, can I get a sum which is better than the individual components? It's very similar to coupling the giraffe and the grasshopper. Okay, right. So what Sukumar Rai told a long time back. So that's what we are going to do. So in Manchester, the graphene came, and over here, from EPFL, molybdenum disulfide came. Can we put them together? We can do this. Can you just press it? There's a video here. We can now make, Yeah, just go on to that. Just press that. That's what it is. So you can do this by simply taking layer by layer. You take a layer of graphene, you can take a layer of boron nitride, you can take a layer of molybdenum disulfide, you can take a layer of metal, you can take a layer of semiconductors, you can just put them together in a stack the way you want, and you can try to make devices. Okay? You can put them together. Okay, so there are many different ways. You can see I can take 40 different materials that are already known today. I can actually take a layer from one, I can take a layer from other. These materials do not exist in nature. I'm making this nature as a three-dimensional metal with a completely user-defined structure. I get to decide what I want, okay? And very good, I have made it. I have put the grasshopper and I put the giraffe together. Can I guess something interesting? Turns out that yes, if I take a layer of graphene and put it on a layer of molybdenum disulfide, this becomes one of the most efficient or sensitive detectors of light humanity has ever produced. So idea here is that you can see that I'm measuring the resistance of the current of the graphene layer with the molybdenum disulfide sitting underneath it. The moment light is on, the current through the graphene layer immediately goes down dramatically. Okay? Now, I can take this current change and divide it by the light, the power of light that I have actually shown. And then it's the basic idea here, of course, is the light is absorbed by the molybdenum disulfide and the electron jumps into graphene, and that leads to a very large current. The idea is that if I convert the responsivity, which is the amount of current I get per unit power of light, it's now the highest sensitivity detector of light in the world. Okay? And you can see the number is ampere per watt is 10 to the power of 9 to 10 to the power of 10, which means that if I have one photon, I told you from the beginning, one photon energy is 10 to the power of minus 19 watt, or joules, right? Per second, it becomes 10 to the power of minus 19 watt. So it means that a minus 19 watt power will cause a photocurrent of 1 nanoampere. And a 1 nanoampere is something that I can measure quite easily in the laboratory. It's no longer 1 femtoampere that most of the other photodetectors are restricted to. It's far more difficult to measure. So you see, this is what I meant. What is emergence here? Graphene does not absorb light, very, very little. Molybdenum disulfide is a semiconductor. It doesn't conduct electricity. It can absorb light, good efficiency, but it doesn't conduct electricity because it's a semiconductor. When I put them together, I have got a conductor and I have an absorber, and I have put them together and give the emergent property of one of the strongest or highest sensitivity optoelectronic material that has ever been produced. Okay? So we need such kind of ideas. Now, this is certainly one of the possible ideas. I have about another 2 minutes. 1 minute? Okay, fine. I'll finish up in 1 minute. Okay? Very, very fast. I had to say this because this is a very, very interesting topic today. It's about brain electronics interface. You know, it's a completely, I wanted to give you, you see, in the emerging technologies, I have quantum technologies, I gave you how a property of matter can be engineered through emergence that can lead to quantum technologies. AI, artificial intelligence, big data, they require computing power, and one of the directions is neuromorphic computing, in which we need to create a brain. Of course, they're very far away. We have got a 10 to 100 billion neurons, about 100 trillion synapses, okay? And the power consumption is 20 watt. If I take the same kind of numbers for silicon MOSFETs, it will be about a megawatt power, which is not possible, right? So we need to create a brain, and can we create a synapse, the basic unit of a brain? A synapse is essentially, I'll go very fast, but you know, basic idea is that I need to create new kinds of memories for if I want to look at brain. And a new kind of memory is that if I keep on, if certain causality you want to strengthen, then you want to create, train the synapse by pulsing, the conductivity should go up. And if I don't want the synapse to work, and you want to deactivate that, then I would like to give opposite pulses, and that should reduce the conductivity of the synapse. So this is what a memory is. This is how we train, right, in a neuromorphic system. So can I make a memory using, at very low power for this kind of application? Turns out that the same set of system which made the highest sensitivity for optoelectronics can be used to create a memory of extremely low power. For example, you can take molybdenum disulfide, graphene like this, put an insulating layer and put a molybdenum disulfide on top, and it turns out that by pulsing this graphene or gate here, we can change the electronic properties of this molybdenum disulfide to act like a brain or a synapse. I don't want to go into that detail, I'm out of time, but you can see that what we can do, you can put pulses like what electrostatic pulses on the gate, and that can, positive pulses reduce the current, and when I put negative pulses, it increases the current through the graphene, and it stays there, which means essentially you can program a synapse like what the synapse does with an electronic device. The advantage of this is my last slide, you can see that using this kind of electronic analogs of synapses, you can measure or you can make synaptic devices whose power consumption is orders of magnitude lower than what the brain does. Typically, a brain takes about few picojoules, about 20 picojoule if I'm not wrong, or 20 femtojoule for every signal processing in the conduction. Whereas you can make, in principle these days, electronic analogs of brain which can be much, much lower in energy. That's it, I'm done. So it's my summary, the performance enhancement for the existing technology and hence go to the new disruptive functionality needs new physics and device concepts, and designing emergence with bottom-up material assembly on devices is a new route. My last statement, you see, when in 2015, there was a statement from Intel saying that what is going to be a smart sensor, a smart city? What's really that we want in the next decade by 2025, which is not too far away from now? Create a device which is no bigger than a grain of rice that can sense, compute, and communicate without wires or maintenance for 10 years. That's the technological challenge that actually can make a serious impact in human life. So what many of our work are geared towards is to make such tiny devices which we can engineer and make them communicable, make them have the computing power on site, and of course various kinds of sensing capabilities. That's the overall goal of the field is. Thank you.
Any questions for sir? Okay. So no questions? Thank you very much, sir. May you kindly take your seat in the audience, please?
Oh, okay. Yeah, sorry, go ahead.
Thank you very much, sir.
Okay.
May I now kindly request our registrar, Ms. Shorbani Shaha, to please present the vote of thanks.
Good afternoon, everybody. While we are approaching towards the end of this program, it is time for me to offer my vote of thanks. Just to wind up, one more time I am telling you, this Science Day, it is a celebration of science. It is celebration of discoveries in science, and for IACS it is special because it is celebration of discovery in our place, that is IACS, where you are visiting. I hope you all enjoyed. You could get glimpses of what we are doing in the laboratories. So we will be happy to welcome you on many more occasions in the future. Feel free to visit IACS to interact with the scientists. They will also be very happy to connect to you. To offer my thanks, first of all, our gratitude to our director for keeping faith in us and giving us the freedom in organizing today's event. Special thanks to Professor Debushri Ghosh, the chairperson of the organizing committee. I said special because she has been bearing with us with our never-ending demands of chairing in this committee and that committee with very diverse composition. This time we had a new committee. The committee did very well. I feel so happy. Thanks to the outgoing executive secretary, Mr. Amit Mujumdar, and also for involving Deepanita Chakraborty, who will be taking over as a new executive secretary. Thanks to all the committee members. They were full of ideas and energy, and we got proof of that they had enormous energy. Many thanks for organizing it in a perfect way. Thank you all.
Thank you very much, ma'am. So with this, I would request you to kindly have a seat for a few more minutes. With this, we come to the conclusion of the program. And it has indeed been a celebration of science. Thank you very much once again for your enthusiastic participation. And lastly, we would be having some refreshments which would be served in the open air theater where you had previously gone for lunch. So you can please head over to the open air theater. Thank you very much once again. Thank you.